And it's a 15-minute presentation, is that right? It's a 15-minute presentation?
Well, I shortened it to 2 hours. Yeah, it's right there.
Okay, all right, okay. Sorry, this always happens after lunch. No, you will not. I know that. Well, good afternoon. I have the pleasure and the honor to introduce our keynote speaker, Dr. Chris Austin, who, as George told you this morning, is the director of NCATS, which is the National Center for Advancing Translational Sciences. Chris is a Princeton grad where he studied biology, and then he spent the next decade or so at Harvard. He went to Harvard Medical School, trained in internal medicine and neurology at Massachusetts General Hospital, and then did a fellowship in genetics at Harvard. He then went to the pharma giant Merck, where he worked at the interface of discovery, genetics, and neuropsychiatry, and particularly focused on schizophrenia. And he was at Merck until he joined the NIH in 2002, where he has been over the past now 14 years. Chris became NCATS director in 2012 and has really initiated and led a number of innovative programs, ranging from the chemical genomics program to predictive toxicology called Tox21, to looking at repurposing drugs for rare and neglected diseases called the Therapeutics for Rare and TNRD, or Therapeutics for Rare and Neglected Diseases program. And most recently has led the effort with Petra Kaufman in establishing throughout the CTSAs a trial innovation network. His scientific leadership, a number of innovative programs and accomplishments have been acknowledged, and he was recently elected to the very prestigious National Academy of Medicine in 2015. So please join me in welcoming Dr. Austin to the podium for his lecture entitled Catalyzing Translational Innovation. Chris?
Well, thanks. It's really great to be here. I have a lot of old friends here and some friends that are relatively new. I have to mention that Vivian Lee and I were in the same National Academy of Medicine class. I don't know if she's still here, but so that was wonderful because we got to know each other through Carrie Byington and Will in their roles in the CTSA program. And I've known George for a very long time through my old job at the National Human Genome Research Institute, and then more recently he's been on our Center Advisory Board since its inception, and it's really helped us get the center started and off the ground. And we're losing him in January, I think. But you know, all good things must come to an end, and so we really appreciate all of the partnership from George and Will and Carrie and Vivian and many others here. So what I'm going to try to do is give you kind of a whirlwind tour, if I can. Yeah, here we go. Of let's see. Let's see. I can do this. Here we go. Yeah, I did that. Doesn't like it. Why don't I just do this? Can I just do that? Yeah. There we go. I'm going to give you a whirlwind tour through some things that NCATS is doing, but I thought I would give you some news from the front on two things that we've been talking about a lot today. If you notice, I've been writing and rewriting my presentation from the beginning. I've been sitting down here on my laptop from the beginning, sort of bringing in and taking things out, depending on things that I think that would interest you. So I'm going to start out telling you a little bit about the moonshot and the PMI initiative, and then I'm going to get into NCATS, and I think it actually will flow pretty well. So, okay, so first the cancer moonshot. I think you all probably know where this came from, but these are slides from Dina Singer, who's at the NCI, is one of the folks who runs this. Obviously, this came out of Joe Biden's leadership in really trying to change the pace of development in cancer, as he likes to say, accomplish 10 years of work in 5 years. Including prevention and screening. And there's a big emphasis on greater cooperation, collaboration, including data sharing. It's always entertaining to watch people who don't know how difficult it is to get scientists to share data, to realize that that's how the world works currently. I was once quoted when I didn't realize that reporters love juicy quotations. I actually was quoted on the front page of the Wall Street Journal saying that scientists will share data only when you pry it from their cold, dead fingers. And so I'm a little careful, more careful when I talk to journalists now. So the Cancer Moonshot panel was, of course, there's a blue ribbon panel that came in to advise the Vice President and the task force, the White House task force, the Vice President's office task force on what should be done. And you see the three co-chairs there. And I just want to highlight Mary Beckerly, who was on this task force as well. And they came up with a group of recommendations that I'm not going to go through. This is all on the Moonshot website. If you just Google Cancer Moonshot Blue Ribbon Panel or anything like that, you'll find them. But if you'll see some themes here that have been coming up today: patient engagement, and you'll hear me talking a lot about that too. Trying to, this came up as well, trying to really organize perhaps all the fantastic work going on in cancer immunotherapy, which just exploded over the last 10 years. Therapeutic drug identification to overcome drug resistance, including how to better do clinical studies of combinations. This is an interesting one. It's a broad term, which essentially means the people who are going to share data better. Looking at fusion oncoproteins in pediatric cancer, symptom management, precision prevention, which was actually the title, as you know, of the previous session before lunch. Taking retrospective analysis of biospecimens that have been treated with standard of care. Creation of a tumor atlas and new enabling technologies. So what are the next steps? There's some of them you'd imagine that the Vice President's Moonshot Task Force, which is based at the White House, had got this report just a little bit ago. Importantly, for this meeting, they identified a number of policy issues, most of which have been talked about already today. How to cover, how to reimburse drugs that are made for segmented group of people, which tend to be a lot more expensive. How to do uniform, flexible, informed consent. Another thing that's come up in the PMI initiative. And of course, NCI now has to figure out what it's going to do of these recommendations and to what extent and in what order. And of course, the extent and rate are going to depend on the appropriation. A bit of good news is in the 21st Century Cures bill that was passed by the House yesterday and will go to the Senate next week, includes a good amount of money for the Moonshot and for the Brain Initiative and for PMI. It's a little different than the mandatory spending that was originally being considered, but it still should do the job. So that's very encouraging. The signs are, although you never know until the the bill is signed by the president. But it looks like the Senate will pass this as well, and the president has been very clear that he wants to sign this as soon as it's passed by both houses. And there's not surprisingly been a lot of emphasis on public-private partnerships, working with companies who make a number of these drugs. Okay, so now I'm going to switch to PMI. What was the artist previously known as the PMI cohort program is now not known as the PMI cohort program, because some community engagement showed that if you go to any place outside of Cambridge, Massachusetts, and you ask them what a PMI cohort is, they amazingly don't know, which was astounding. So they changed this, given the patient partnership that is a hallmark of this program, into the All of Us program. Now, it's good and bad, right? I mean, the PMI cohort at least tells you that it's about precision, it's about medicine, it's an initiative. Nobody knows what a cohort is, but you could probably explain it. All of Us, of course, could be almost anything, but this was actually the name that was suggested by the patient group, the participant group, and it's now been accepted. So it's now called the All of Us Research Program. So, of course, the point is to build a large infrastructure of 1 million or more Americans that will act as a platform for scientific discovery. And this is a lot is focused on genes, but of course it's not just about genes. It's about clinical information, electronic health record information, environmental information, lifestyle, and genetic data for 1 million participants. And importantly, this is going to be a longitudinal study. It is projected to go for at least 60 years. One can imagine this turning into a massive version of Framingham, which has been going on about that long, a little bit longer. Importantly, it's not a representative study from an epidemiological standpoint. A lot of discussion went into this, but it is about this issue. It's a very important issue. But it is meant to reflect the very broad diversity and really unique diversity that the U.S. has across not only race and ethnicity, but about gender, socioeconomic status, geography, health status, and age. And if one looks, and all this is on the website, if you look at their recruitment strategy, it's actually to overrepresent, to oversample in groups which are traditionally underrepresented in biomedical research. So that's a very intentional strategy from the very beginning. And a really interesting thing that has been a characteristic of this from the beginning is to build the tools that are necessary, including modern mobile computing and iPhone or smartphone apps, to be able to deliver information in real time back to participants. That's been a sort of bedrock issue from the very beginning. And you probably know that there are two pieces of this cohort. There are large health provider organizations, which are thought to be, and I'll show you some of those in a minute, are thought to contribute probably two-thirds or so of the number of 1 million. But one of the things that the president said from the very beginning, which is very challenging, is that he wanted anybody in the country, no matter where they are, to be able to raise their hand and say, I want to participate. And that, of course, is non-trivial, as all of us who've done clinical research know. But it is something which I think is really going to mean that this is a different kind of research program run by the participants, not necessarily by the doctors. And that's actually being done intentionally. Okay, so here's the core values. As I just mentioned, it's participation open to anyone. Focus on rich diversity of the American population. Participants are never going to be called subjects. They're going to be called partners or participants. And that has been a very large cultural part of this program from the very beginning. And as part of that, participants are going to have access to information, to the information that is generated on them individually, as well as in larger meta-analyses or analyses of people like them. One of the reasons this was done was that if you think about retention in a study like this over 60 years, the only way to have a study retain people is to give people something back. And traditionally, clinical trials will pay participants, but that's just not. It has all sorts of perverse incentives associated with it, but it's also just not scalable. So what people really want when they were asked, and there was a lot of outreach that was done at the beginning, what people really wanted is they wanted to contribute to a national initiative that would help their fellow human beings, but they also wanted information on themselves. We're all kind of neurotic about checking our weight and our heart rate and our Fitbits and all this stuff. So if you can imagine that you would get all of that information in really an expert way by contributing to this, contributing your information, that's very appealing to a lot of people. Of course, that creates major issues of privacy and trust. Another person who's joining our council, Matt Might, you probably remember, has been working at the White House on cybersecurity for this initiative from the very beginning. And of course, that's the assumption is this is going to, once it's out, it is going to become almost overnight the biggest target for hackers worldwide. That's the assumption. So it's being designed in as careful a way as it can be. These are the four components. There's a data coordinating center, essentially the data and research support center. There's a biobank at the Mayo Clinic, a participant technology center, which means particularly around the volunteer participant center, because it's important that if you're going to have populations of people who don't usually participate in research, some of these people do not have internet access. They don't have a smartphone. They may have a regular phone, but they may not have the tools that all of us take for granted. And then there are, of course, the large health provider organizations, including the VA that we're going to hear about in a minute, but these regional medical centers that I'll show you, and these federally qualified health centers, which take care of a lot of the lower socioeconomic groups in the country. So the major building blocks, such as they are, a lot of this is building a foundation for a program that is going to be going on for a long time, we hope. There's an awarding network of over 50 awardees, and I'll show you those on the next slide. There is a draft protocol of the IRB that's a central IRB being run out of the NIH centrally. Number of workshops that I mentioned already on community outreach, and a lot of how the program is designed came from the results of those community outreach efforts. They had the all-important 1-800 number and some smartphone apps and a data center, which have been developed. They're not quite ready to go yet, but they've been developed, and biobank IT's capacity, et cetera. So these are the groups that are involved now. On the bottom, below the chevron, are the academic groups or academic or health providers, including some academic places like Columbia, Northwestern, Arizona, Pittsburgh, and some health provider organizations like the Kaiser Health System, the VA, et cetera. And then up here are some of the IT direct volunteer work through Eric Topol's group at the Scripps, but also places like Walgreens and Blue Cross Blue Shield, SAGE, which a lot of you know, so all of these are part of a very large collaborative network. Eric Dishman somehow keeps all these people straight. Eric Dishman himself is a very interesting person. He's the one who's been recruited to run this program. He's a social scientist by background. He's not a geneticist. He's not a clinician. He's a social scientist who then went to Intel. He spent the last 12 years at Intel working on large health systems and informatics for large health systems. And interestingly, in the midst of this, got a rare kidney cancer that was amenable to a targeted therapy and is alive because of precision medicine. So Eric is a walking example of the promise of precision medicine. But as we were talking about this morning, the question is, how do you generalize that? And so that's what I'm going to spend the rest of my time talking about. Okay, so Here's a slide. I'm hoping to this is my transition from those two into what NCATS does. So I think a lot about rare disease medicine, and it's interesting to think about some of the dynamics that we were talking about this morning, and to realize that many or most of the issues that are being discussed with some fervor in the personalized medicine community, and as if they are new, are absolutely old hat to the rare disease community and have been from the very beginning. And so personalized medicine, a number of people have made this point. The doctors have been trying to do personalized medicine for thousands of years. This is just being done on a more systematic level. But happily for us, we're not starting from scratch. So if you think about this on sort of two axes, think about population first. So if you go from population from the top to the bottom, prevalence going from high to low, at the top, they're common diseases, and I'm going to use the definition of the Orphan Drug Act from FDA, which is less than 200,000 U.S. prevalence is a rare disease. So everything above that is a common disease. So rare disease is 200,000 or below, and then we might call a personal disease, truly personalized medicine, which is one person and of one medicine. Now, there are great examples of this. Now, the example that I like using, that everybody likes using, is crizotinib, right? So the U.S. incidence. Now, I'm going to talk about two different things. Incidence for cancer, prevalence for rare disease. All lung cancer is about 200,000, maybe 250,000 a year. And that is treatable, not terribly well, but it is treatable by cisplatin. And that's been around forever. It's about $20 a dose. Okay, so ALK positive lung cancer is about 3, 4, 5% of all lung cancers. So that's about 5,000 U.S. incidents. So that is clearly a rare disease. It's about the same as a rare disease I picked off the shelf called Hunter syndrome, one of about 6,000 genetic rare diseases. Prevalence of about 2,000. And you look at the prices of these drugs, crizotinib being $13,500 a dose, and Elaprase, which is a enzyme replacement therapy for Hunter syndrome, is $375,000 a year for the life of the patient. And these are pediatric onset diseases. So this is like buying a house in a relatively expensive city every year for your entire life. Now, I did a calculation a few years ago. If we had a drug for every rare disease patient today, and we priced it at a relatively bargain rate, which is $200,000 a year, drugs for rare disease patients alone would consume one-half U.S. GDP. One-half the U.S. GDP just to pay for drugs for rare disease patients. So in other words, don't bother, right? The scale is completely off. So realize that there's some great examples here. Crizotinib's wonderful. Having Elaprase, if you have a child with Hunter syndrome, is wonderful, but it is completely non-scalable. So we at NCATS are constantly asking ourselves, how do we not only improve, but how do we do it in a logarithmic way? So I come from the Genome Institute. I'm a genome guy by background, and somebody showed the sequencing Moore's Law comparison, right? So the reason that genome sequencing has become so much more efficient is the cost of genome sequencing has come down 10,000-fold in the last 15 years. Now, we're probably not going to get that with drug development, but the point is we can't go for 10% change. We have to go for 10-fold, 100-fold change. Another number I'll throw at you of the diseases that are out there that are untreatable. At the current rate of diseases going from untreatable to treatable, it will be 2,000 years before every human disease is treatable. So that, I would submit to you, is not an acceptable answer. But it also tells you that we can't do this with incremental improvement. Okay, so if you get down here, it gets even worse, right? CAR-T immunotherapy, I've seen some estimates from Carl June that might cost $500,000, $750,000 a person. There's a gene therapy in Europe which costs $1 million a treatment, and realize that these are becoming more common via things like the Undiagnosed Disease Program that are identifying diseases for the first time, N of 1. And one example, one of my favorites, is NGLY1, which is actually the disease that Matt Might's son has. So realize that rare diseases are trying to find more people. You get an N of 1, try to find more people. Maybe there's 50, 100, or 200 that have your disease. And in common diseases, they're being segmented. But the bottom line is they're all coming into this rare disease space that's exceedingly difficult. for any number of reasons. And what I'm going to focus on for the rest of the talk is how do we at NCATS think about how to change the rules of the game so that interventions can be developed much more rapidly and efficiently, and the cost will be manageable. And the bottom line here is that we're trying to decrease the cost of production. Essentially, is what it comes down to. Okay, so the good news is this. I'll show you what the good news is in a second. So I graduated from medical school in 1986. That's down here. Well, actually 2 years before this. And at the time that I graduated from medical school, there were about a dozen human conditions, the molecular basis of which was known. About 10 or 12. Now, over the last ensuing 30 years, that number is well over 6,000 now. There's still probably 1,000 diseases, the molecular basis of which we don't know, but that is an incredible change. However, remember that the first genetic disease to be defined is sickle cell anemia. Sickle cell disease in 1949, there's still not a drug directed against that molecular pathogenesis. So just because you have the gene doesn't mean you're going to have a drug. There's this interesting fantasy that particularly people in Congress have, that and a lot of scientists and a lot of doctors, that all you got to do is have the cell paper announcing the gene, and as the night follows the day, there will be a drug. As Will knows better than anybody, that is not the case. It's actually quite the opposite. And that's manifested by this, that only about 500 of these have therapy. And as a neurologist, these are the patients I used to see. Patients with ALS, Huntington's disease, Alzheimer's disease, etc. Okay, so this is the problem. We live in this paradoxical, bittersweet time where we know more about ourselves in health and disease than we ever have, perhaps exemplified by the Genome Project, but there's lots of examples. It's fantastic science. However, if you ask, is healthcare, for the most part, healthcare, is the practice of neurology, for instance, any different now than it was 30 years ago? The answer is pretty much no. You know, I went to the doc probably 6 months ago, and the experience was pretty much what it was 30 years ago. And there's this wonderful IOM report called Shorter Lives, Poorer Health that goes into the consequences of that mismatch. And so why are we so bad at going from this side to this side? You know, there's all kinds of knock-on effects of this. Bottom line is that people are unhealthier than they should be, and funders a long time ago in the public sector and the private sector began to get cranky with this. And this is not just Congress, but this is also the private sector, which has become quite impatient. So our hypothesis here, and our working hypothesis at the starting of NCATS, was that this is a scientific problem that needs to be addressed as a scientific problem, and only when it is understood as a scientific process will translation be turned from an empirical, phenomenological process into a predictive science. And only when it's a predictive science will success rates improve and costs come down. Okay, so that leads me to the question of what the heck is translation? So NCATS is really behind the eight ball in the sense that it's the only NIH institute that has a word in the name that has some non-medical meaning, right? The National Cancer Institute does not have that problem. The National Eye Institute does not have that problem. To normal people, translation means linguistics. Right? So we always have to define what we're doing and then tell them how we're going to solve the problem. One of the biggest problems I've had as director is actually convincing people that translation even exists. I mean, there's many people who think that there's a basic science discovery, and then there's pharmas that put ads on TV and charge large amounts of money from the drug that the basic researcher made, and that's a direct transition. They really believe this, and they make policies dependent on it. So I spend a lot of my time in telling people about the fact that no, there is this very large swamp, 10 or 15 year swamp called translation in the middle. So what is translation? It's the whole process of going from an observation that a scientist makes in the lab, a basic researcher by training, or a doc makes in the clinic, or a community health worker makes in the community, think about Zika or Ebola, into an intervention, a drug, a device, a behavioral intervention that is shown to improve the health of individuals in a clinical trial, and then the public, right? As a friend of mine likes to call us free-range humans, you know, people who are out in the community, you know, eating and drinking crazy things and doing things they're not supposed to. But you notice that NCATS is not the National Center for Translation. Virtually every institute at center at NIH does translation to some degree, given this definition. Every biotech, every pharma, et cetera. We are the National Center for Advancing Translational Science. Right? So we're trying to make something better, advancing translational science. What the heck is translational science? Well, this is a new field. It's never existed before. And we define it as the field of investigation that seeks to understand what the general principles are of each step of the translational process. Now, any of you who've done translation know that it is a painfully empirical process. You know, if you ask a medicinal chemist, how do they know how to change a compound to make it more potent? You know what they say? Intuition. Really? And that's viewed as a positive. You know, intuition. Man, data. You know, intuition is what witch doctors use. Astrologists use intuition, not scientists. And this is true with toxicology. It's true with clinical trials. It's true. You know, where I was trained at the August Mass General Hospital, I was actually told by one of the people there, bringing them data, one of the professors, and I showed him all this data. He said, "Why aren't we doing what the data shows?" He said, "The data don't matter. This is the Massachusetts General Hospital. And here, this is what we do." You know, so, this is both a scientific problem and a cultural problem, and an organizational problem. Okay, so then people say, well, okay, what do you guys do? So if you don't have a disease, you don't have an organ, what is the process you work on? Our disease is the translational process, and our patient has a problem list. This is the translational patient's problem list. These are the problems which bedevil translation. I don't care whether you're working on psoriasis or Alzheimer's disease, it's the same problems. The first three are the big three reasons that drugs fail. The others, preclinically, the others tend to be clinical. So many things that are massive general problems. But there's also a lot of organizational things that we work on, which you've been talking about today. Data release, IP management, project management, credit for team science, huge problem with academic centers. Incentives for health improvements, education, training, collaborative structures. These are all things that are every bit as common at causing translational failure as the scientists. If you look at this, translational failure is about 50% caused by scientific issues, 50% by organizational issues. So we work on those too. Okay, so just a few examples, and I'm just going to give you these as examples of the kinds of things that we're doing, because of course I don't have time to tell you about everything. It was talked about earlier that maybe a problem with finding cancer interventions or actionable mutations is that there are not drugs available for those mutations. That's absolutely true. It's also true with rare diseases. I just showed you. So we and a lot of other people had this idea, well, just do drug repurposing. Why would you do that? We and others, if we're looking for a novel compound, a novel drug, we have to start from scratch. We start out with the usual 500,000 compounds, takes 15 to 20 years to go from the left side to the right side, fails about 99% of the time. And so the concept was, well, if we could put every drug ever approved for human use into a collection and then screen them across human-based assays, could we repurpose them in a relatively short time? And believe it or not, when we started doing this, there was no complete non-redundant list of every drug ever approved for human use. And you must say, you've got to be kidding me. Nobody ever put together a list of every drug ever approved by the major markets. I'm talking about Europe, US, Europe, Canada, Japan. Never been done. So a lot of reasons, this is not a scientific problem, it's an organizational cultural problem, incentive problem, we could talk about if you're interested. But we did this. So we finally took our informatics people and our compound management people about 5 years, kind of reads like a John le Carré novel if you want to read it, why it took so long, but it's now done, all the data's out on our website, you can download it if you want. But one of the things that we've done, we do many collaborations with people all over the world to take advantage of this collection. But one of the things that we've done that's really improved efficiency also, after we had this whole collection, was had patients and foundations coming to us to use this collection. So this one is each one of these happened because a patient or a parent walked into my office and said, can we work with you? They're not scientists. This happens to be on Charcot-Marie Tooth disease. It's a rare neuropathy. This is a rare cancer called chordoma, and this is a rare muscle disease called myotonic dystrophy. In each case, this was driven by the patients who then nucleated a science group, not the other way around. And it actually was more efficient. We also are doing this via our SBIR program. I had this in here already, but where's the recursion guys? Are they still here? Yeah, they're there. So this is an SBIR grant that we award that we were really happy to approve for a company called Recursion, which is right across town, which has a really impressive innovative platform for doing massive repurposing for rare diseases. There's a video on our website if you don't want to go across town to see what they have. And this is all from our website, a really long story on our website about this. And this is to prove to you that these people are actually in Utah, because you can see back here in the bad weather, and it's snowing like it is today. But the point is that we're attacking this from multiple avenues, including the company avenue. So let's say you have a novel target, but you run the repurposing route and you don't get anything. Then what do? Well, you have to drug undruggable targets. About 90% of the human genome is undrugged. It's not a small molecule. These things are too risky for private sector to take on in general. So we do a lot of de-risking of novel targets. This happens to be on something called one of the aldehyde dehydrogenases. Again, everything we do is a collaboration. So this is with Bacillus Vacilius, some of you may know, who's at Yale now. There's actually 18 ALDHs, and this happens to be on 1A1, which is important for maintenance of stemness. So as you might imagine, it's mutated and dysregulated in cancer. And Bacillus, who doesn't know anything about drugs, came to us and said, well, can we develop this with you? And we don't know anything. Our guys didn't know anything about aldehyde dehydrogenase either. But in the course of about 2 years, we went from that to drugs which can be tested in animals, which they are now, with all the usual, if you're a drug person, all the usual kind of PK characteristics you'd want, and a lot of nice publications. So we do a lot of this too, probably 100 projects going on at any one time. Let's say you're successful at this stage. If it's for a rare disease, a company still is not going to be interested, so then you have to de-risk it to the point of usually an IND or a phase 1, phase 2. And so we have a different program that Will mentioned, Trend, and another one called Bridges that does this. So it's, again, same model, collaboration between drug developers and NCATs and outside investigators with disease or target expertise. About half the time these are companies, half the time they're academics. And the point is to get this, not a publication, the point is a license to a company that will take this off our hands and develop it through approval and marketing. And I just show you this to show you that if you're an academic investigator, it is very hard to do these things on NIH funding, all of these lists. Some of these things you may not even know what they are, and that's kind of the point. But that's okay, because our people don't have the disease expertise that you have. And so together, that's how we manage to do these things. Lastly, I want to tell you something about one of our efforts to improve drug development effectiveness. So we have programs that address all of that whole list, that whole problem list that I showed you before. A big problem of drugs getting into people is that despite looking safe and efficacious in an animal model, generally a rodent, they are either not safe or not efficacious in the human. Now, I tell my mother that, and she says, like, duh. You know, I mean, my mother seems to know that rats are not humans, but scientists generally, not so much. You know, we've been brainwashed into this idea that a mouse is an animal model, and that model is going to be predictive, right? And so it's always interesting to work with people who say, what were you thinking? But that was done for reasons that I think we all know and understand. But the question here is, given the fact that 90% of drugs which go into people, phase 1 trials, never get approved. So 90%, even when you get into people, so you can do 10 years of work, lots of things fail there. Even when you get to the first in human, still another 90% fail, right? So how do you address that problem? Well, the question is, taking advantage of stem cell technology, microfluidics, tissue printing, and cell sensor technology, bioengineering, could we create little human organoids, tissue chips or microfluidic systems, which would represent the structural and functional elements of all human tissues as a way to test safety and efficacy of new therapeutics. And so this, like NCATS does, everything is a collaboration, and this has to be a collaboration with DARPA. This is the Defense Advanced Research Projects Agency. This is the Skunk Works part of the DoD, brought you the internet and the GPS and a number of other things, and the FDA. And we spent the first couple of years developing each individual organ, and here are the individual organs. This is from our website. Yes, this gentleman's name is Chip. Sorry about that. But he has all these organs here, and if you click on them, you will find microfluidic platforms developed for each of those organs. And then, of course, Chip is not individual organs. He has all of his chips, all of his organs connected, so we needed to connect all of these in a modular platform, and so that we've been doing since then. We're now at 8 chips, 8 organs connected, and probably by the end of next year we'll be at all 10. And where we are now is, as I mentioned, integration, compound testing with canonical compounds, and now live compounds from the IQ Consortium and pharmas, validating these via external validation centers. I don't know if Carrie's still here, but Carrie, are you still here? Oh, there you are. Yeah, one of these is at A&M, the validation centers. And the idea here is if these are going to be used by the community, they have to work in somebody's hands who had nothing to do with creating them, right? And you all know what the issues there have been. So there are 2 centers which are going to be doing target tissue chip validation, partnerships, particularly with pharma, and then how to disseminate these so that people can use them. And this is the consortium. There's a massive consortium here run by us, FDA, and DARPA, but you notice all of these different academic centers which are working on different organs. They work very closely with each other, but they're separate organizations. And then there's a lot of biotech and pharma partnerships. And I just want to show you this one because it's an interesting sort of cancer example, if I can make this work. This is tumor vasculature. I don't know if it'll work, but yeah. So this is made from a microvascular network made from iPS cells made from tumors. And so what you notice here is that these cells are coming down here through the normal vasculature, go through this really screwy tumor vasculature here. And this is actually what it looks like. This is colon tumor made from this ACT116 cell line, supported by this perfusing vasculature. So and there's I don't have time I could give you another hour of cool videos and things. But what I would urge you to do, if you're interested, just go to the website. There's a fantastic website with videos of each of these organs: liver, kidney, brain, etc. And so where are we going with this? And this is one of the reasons that I'm so excited about translation and the potential here is that things are happening much faster than I would have anticipated. And why is that? I think it's because we take a relentlessly team approach to everything. We believe very strongly that translation is a team sport, needs to be organized that way, and if you bring people with many diverse experiences and expertise together, magic things happen, and indeed they do happen. So we are now 5 years after the start of this program considering this, doing what we're calling U on a chip. Because these are made, for the most part, from iPS cells, it is as easy for us to make 200 chips of the same person as a chip from each of the people in this room. Now, think of the implications of that. Now, we're not only not testing in a generic syngeneic mouse, we're testing a human who are outbred, and we're not even testing, say, in a phase 1 trial, a random group of people. We're able actually to make individual chips from individual people and test safety and efficacy in individual people from the very beginning. And so we're now planning a clinical trial on chips that we're going to be doing with the CTSA program, where we work with, we do a clinical trial in people and a clinical trial in chips made from them at the same time. I do want to tell you 2 other things. One about this and one about the CTSA program. So let's say you've got an intervention. How are you going to test it? Well, with a rare disease and with a common disease with molecular profiling, of course, you got to find the patients, and they're actually quite rare. And so we have, within the rare disease program, we have a clinical trial a clinical network made up of 22 different consortia of different groups of rare diseases organized by a particular biological principle, an organ, an organelle, etc., each of which has a patient advocacy group associated with it. And what we're finding is that by grouping rare diseases and studying them as a group, and by involving patients in the very beginning, the headway, the progress is greatly accelerated. And this is allowing us to identify patient cohorts for potential clinical trials, doing registries, natural history, all that kind of stuff. So lastly, and this really is the last thing I'm going to tell you, on the CTSA side, so a real issue with doing clinical studies in this country is even if I handed Will or Carrie or Vivian or anyone a treatment for every untreatable disease tomorrow, we in no way have a capacity in this country to test them within any reasonable period of time. And when I took over NCATS 4 years ago, I looked at the CTSA map, and I looked at the PIs, who include, if you can imagine, 60 Carrie Bynstons. These are really smart people. And they are surrounded by people like Will and the Will and Carrie equivalents at most of the best academic medical centers in the country. But they had not been incentivized to work together. Actually, it was very hard for them to work together, given the way NIH had been managing. So we thought, well, gosh, just imagine if we could apply this team concept of people working together and sharing experience and everything from training to doing clinical trials. We could really do some serious damage on this problem. And so that's what we're doing. The CTSA program, as you probably know, is a national consortium of medical research institutions, works on the lower part of that problem list I showed you at the beginning and does a lot of training. And one of the things we started doing with the PIs at the very beginning, starting literally a couple months after I became director, was to say, well, what are the principal roadblocks in doing clinical translation efficiently? And it occurred to us that what we really needed was, and these words are taken intentionally, an efficient, collaborative national network for translational medicine. And I'm glad to say that we're almost there now with this Trial Innovation Network. And we focused on 3 issues initially: IRB, multi-site IRB review, recruitment and investigator qualification, largely solved those problems, and we're working on other things now like contracting and budgeting and things like that. All of this has been wrapped into something called the Trial Innovation Network that we're very excited about, and it's going to be launched shortly. So this is what the map looks like. In case you're wondering if you got lost, you're here. But you notice that this consortium is really quite wonderful. It's east, west, north, south, really rural, urban, really tremendous diversity of places. And interestingly, and I use Utah here on purpose, because when I was out here, I heard this, that Utah has been very progressive in reaching out to outside of Salt Lake City and outside of Utah to Arizona, other states, and many other. Some other CTSAs have done this as well, so they have sort of regional networks. They're not point sources. They're actually regional collaborative blobs, I guess is a nice way to put it. And so the question was very simple. How do we make this a national blob? Right, so that's really what we've done, and if any of you have read, if you haven't read it, I would encourage you to do it. Read the Idea Factory. If you've read this fantastic story of Bell Labs and the creation of the first national phone network. A lot of the same issues that is allowing us to form virtual teams just in time to do a clinical study, share information on all kinds of things, best practices, tools, training, connect data systems via a harmonized electronic health record, implement efficient multi-site studies, and we're aiming for a 10-fold improvement in IRB and recruitment times, integrate care and research. We have this crazy system in this country where most places care happens in one place, research happens in another, and they don't talk to each other. It's quite amazing. And some of the same people do both. And most importantly, have greater impact together. For who? For the patients. So we are very clear that our constituents are the patients, full stop. And I know this sounds funny, but I find it, it is at NIH. Sometimes we lose track of that. We lose track who we're working for and why we're doing this, and we try to keep very much in front of ourselves. You know, if I was a patient with a rare disease or I had a child with a rare cancer, what would I want? And a lot of the decisions that we've made have become much easier when we ask ourselves that question. So we have a lot of information. You can go to our website, follow us on Twitter and YouTube and all kinds of things. There are some things on the YouTube channel that I'm glad Will did not show. There's some videos there that will remain anonymous. But I don't know if we have time for questions. I'm going to be around the rest of the day. Be glad to talk to you. Thank you.
Why don't we take a couple of questions? Anyone questions for Dr. Austin? No questions. Yes.
So I very much enjoyed your presentation. One question I had about the scalability had to do with international initiatives. I mean, you looked at U.S. constituencies. I wondered if you could talk more globally.
Yeah, thank you. That was, I didn't even plant that question. That's a great, so fantastic point. As we become more segmented in the groups of patients that we're looking for, and once we begin thinking that there are thousands of diseases that we have to figure out, and the patients are waiting for treatments for them now, and they hope to get them in their kid's lifetime. This is not a U.S. problem. These are the human family problem. And so we have been very aggressive with NCATS starting international initiatives. We have one on preclinical translation, sharing all best practices, all of that kind of stuff, and technologies and programs with Australia, Europe, Canada, and Japan. Earlier this year, I took over chairmanship of something called the International Rare Disease Research Consortium, which is coordinating all the world's major and now more developing world funding agencies, patient groups, and companies to accelerate by a factor of 10 the progress that we're making in rare diseases by sharing. There's another one at the NIH, which I have less involved with, something called the Global Alliance for Chronic Disease, which Roger Glass is involved in. But the point is that there are, no matter where I go, whether it's in Africa, South America, Japan, Canada, Europe, the issues are all the same. And I came from the Genome Institute that brought you the Genome Project, and if you remember the history of the Genome Project, the only reason that project got done on time and under budget was because it was an explicitly international initiative. From the very beginning, people said the human genome is not a U.S. genome or an African genome or a whatever. It's a human genome, and it needs to be presented. It needs to be approached that way, and it was far more successful because of that. And it's, of course, what it does is it's made things even more complicated because now we're trying to do IRB. We've basically handled IRB harmonization within this country. Now we're trying to do IRB harmonization with France and Japan and all of that, which is more difficult. But you know, we come down and we ask ourselves, you know, okay, so you have a patient, and I had this happen just a little while ago, a child who was 6 months old, was a grandson of a person who's the deputy director of the CTSA program, had a large brainstem glioma. And she was told that there was a drug, it was a MEK inhibitor, that was available, but the trial had been stuck in the IRB for a year at the institute, so she couldn't get the drug. And that kind of thing just makes my skin crawl. You know, we're here, this is a human-created problem that is completely solvable, and people are dying because we can't get our act together to figure it out. And as long as we keep that foremost in our head, then it's very clear we're going to be able to solve this on a national level, too.
Yeah, just quickly, I think, well, first of all, this is amazing. It's very great how you go with team science and very creative about so many different questions. So when I was at the big data workshop at the NCI, the question of consent and IRB
Yeah.
did come up, and I think
Yep.
we're very lucky here in Utah with having a great IRB, but others are not. But
Yep.
also, some people are taking shortcuts now with consents that really do not cover the work
Yes.
that they're doing and the sharing
Yep.
they're doing.
Yep.
So is that something that you're helping the other institutions out with, or
Yeah.
so that there's no reinvention of the wheel, and how can we benefit from the process that you have started?
Yeah. So we once we got these first three done, one of the next ones that we worked on was a consent process, a flexible consent process, but also a machine readable consent process, so that you didn't. As you probably know, if you're trying to look at consents across institutions, frequently these things are scanned. They're PDFs or they're JPEGs, and that's just not scalable if you have thousands of people. And so we're actually, that's one of the initiatives that we're doing, both within the CTSA program and within NERDRC, within the International Rare Disease Consortium. This one is a collaboration with GA4GH, if you know these people, the Global Alliance for Genomics and Health. We also have been working very closely with FDA on this. So there's a harmonization of consent and harmonization of clinical trial templates, which we've done, which is going to be coming out pretty soon. It's the same people who brought you the the biomarker dictionary glossary. It's called BEST, that came out a little while ago. That was also an NIH FDA thing that I could say this here. It's said to be NIH, but we did almost all the work. But I have to give the boss credit now and then. So
And last question.
Yeah, thanks, Dr. Austin. Tim Constantine from Recursion. Full disclosure, we're recipients of funding from NCAT, so we're very grateful for that. I'm really excited to hear you speak today, and I think we fully support the position that there's been a failure of imagination when it comes to how to address some of these issues. I'm hoping you could speak, perhaps, we've talked a lot about technical feasibility, scientific data sharing. When it comes to the overall regulatory paradigm and how you run clinical trials for smaller and smaller patient cohorts, and how do we work best with FDA to get from IND to NDA?
Yeah, so this makes me, this is one of the reasons that I'm most optimistic about this. So when I went to NIH, so I came from Merck, where when I would call the FDA, it was like, it was not a friendly relationship. It was cordial, but clearly we were the enemy, and it was an adversarial relationship. At NIH, it's not that way, because we're on the same email server. We're part of the same department, and that really does make a difference. It's not that if we go with a project that we're working on and we have a collaborator or something, we get treated the same way as everybody else, but we can go to them very easily and say, can you just talk to me? Can you just, tell me what the general problems are? And how might we work together to establish a paradigm so that when we or somebody else comes in the next time, we don't have to deal with the system that everybody realizes doesn't work. And that's a conversation that they really welcome. And the place is very different than it was when I got there 14 years ago. So so every one of the projects I talked about, every single one of them, we have partners from the FDA on our project teams from the very beginning, because we realize exactly what you said. We could do the greatest science in the world, gets the FDA doesn't go anywhere. That's not going to work. I'm particularly optimistic by what's happened to the FDA and NIH Leadership Council. So this was sort of moribund for a while, but it really has taken off in the last 2 or 3 years, doing some very effective work, particularly on the areas of small clinical trials. We've been doing this with FDA for a number of years because of rare diseases, because they're all small. But it's becoming more and more common, of course, with segmented common diseases. And just in case all of you don't realize this, if you think about how precision medicine, how we tend to think about precision medicine, we're going from a phenotypic, a disorder which is identified phenotypically, symptomatically, and then we segment it on the basis of genetics. Rare diseases are the other way around. They get defined on the basis of a genetic defect, and then there is a phenotypic spectrum that's defined on the basis of that. So they really are complementary to each other. But in the end, if you're trying to do a clinical trial, it's exactly the same problem. And so we actually, together with FDA, we've been running symposia every year on small clinical trials, segmentation, all that kind of stuff. We are very hopeful, although I have no inside information about what's going to happen, that Rob may stay, Rob Califf, because he is just a breath of fresh air there and really knows his stuff. He's a former CTSA PI, so he understands all of these issues, has been an incredible partner. But I think even if he leaves, I think he's established a culture there, which I'm actually quite optimistic about.
Okay. Please join me in thanking Chris.
Okay, great. So thanks very much. So why don't we just take 5 quick minutes as we shift over to the third session of the program, which relates to precision medicine and the public. And so, Jim, we can get your panel up here, and then we can. Yeah. We're a bit behind. We'll make sure. Oh, yeah, go ahead.
Schaefer, that's him, or that's Kathy.
Schaefer.
And then Meyer is going to be Larry.
Do you have one?
No.
Okay, so are you going to speak? Okay, if we can get people to take their seats, please. We're going to get the next panel started. Okay. Okay, great. So leading this panel as a moderator, we have Professor Jim Tabery in the Department of Philosophy here at the University of Utah. So Jim, over to you.
Thanks, George. Good afternoon. We've heard several times today the Precision Medicine Initiative mentioned, just most recently in our keynote address. That was announced by President Obama in his 2015 State of the Union address. It was received with quite a bit of fanfare. There was a lot of excitement about the ambitious scope of it, the scale, the size. It also brought attention to the great many challenges that would be involved in trying to enroll 1 million people nationwide, get sensitive information from them, both in terms of their genetics and their health information, and track environmental information from them long term. What received less attention was how many large longitudinal research cohorts are already out there in the U.S. and abroad. And those are very useful because they give us examples of other cohorts and other leaders of those cohorts who have already wrestled with the technological, scientific, ethical challenges that are involved in trying to recruit a large number of people, keep them invested in a project over time, and provide us with sensitive information that's valuable to researchers. So we've got examples where folks have faced those challenges, and they've also overcome them and provided valuable resources for researchers. So the goal of the next session on large research cohorts is to have representatives from 2 of those to talk about their experiences. Our first speaker is going to be Dr. Laurence Meyer. He is from the VA and has experience with the Million Vets program, which aims to enroll 1 million veterans into a large research project. And then our next speaker will be Dr. Catherine Schaefer from Kaiser Permanente. She is the director of the research program in genetics, environment, and health at Kaiser Permanente. And they have already enrolled several 100,000 members of Kaiser Permanente into their research cohort, which is now going to be a resource for researchers. So the plan will be 15 minutes from Dr. Meyer, 15 minutes from Dr. Schaefer, and then we'll have 30 minutes to discuss with them the issues that come up. Dr. Meyer.
So thank you. I am from the VA, but I'm also from the University of Utah, and I've sort of got informal confirmation today that I can actually stay here, even though I'm at the VA. So the VA, a lot of people are confused. There's the VHA, which is Veterans Health Administration, which is what a lot of people think of. It's the hospitals. There's also VBA, think GI Bill, home loans, also all of the pensions, and they control access to VHA. And then there's cemeteries. So there's really 3 main veteran-facing arms, as well as some other arms of OIMT, for example. So IT is all segregated within the VA. But within VHA, everybody thinks of providing health care, which is really important, and the VA, I think, does a pretty good job of that once the veterans actually get into the system. But research and training are also issues. Well over half of all medical professionals, and that's not just physicians, that's pharmacists, nurses, physical therapists, respiratory therapists, social workers, mental health, across the board get their training in the VA. And then research. So we are an integrated system. There are 152 big VA medical centers, well over 1,000 points of care. And of those, 120 of the VAs are affiliated with academic institutions, as the University of Utah is affiliated with this one, and about 100 of those do research. There are the number of individual research programs that are going on, focusing specifically on diseases of veterans. Diseases of TBI, PTSD, rehabilitation medicine. There's a whole section on rehab medicine, all the way to basic biomedical research is really large. We also are blessed with one of the first electronic health record systems in the world, part of which was developed here in Salt Lake City. And it used to be an old, C cursor flashing up there at you, and now it's really developed into a very nice GUI front end. That is about 10 years old, and it's just going to be redone. But we have very solid 20-year data on almost everybody, and we have 30-year data going back on a lot of people. So we have records on almost 20 million people. Then we can also, in part, not perfectly by any means, but we can cut short that 10 to 15-year cycle of translation, since we have this captive audience of physicians. So about 10 years ago, there were those of us that were interested in genomics, and they said, should we do a genomic project? And so they did something very smart, and I wasn't part of this. They reached out to veterans and said, are you in favor of this? How should we do it? What should we do with it? And so this was called the Veteran Consultation Project. Kathy Hudson, who's now at the NIH, actually was the lead on this, and she went through VSOs, the Veteran Service Organizations, which means American Legion, Disabled American Veteran, and then there's a host of the new groups now. So there's a lot of these VSOs, as well as internet surveys, and said, what shall we do? So initially they asked, do you think we ought to do a genetic project? In round numbers, 80% said yes, and then we said, well, here's what we're going to do, and at the end of it, another couple said yes. So veterans were very in support of this. They asked a whole lot of sub-questions. I'm not nearly going to go through them all. Participating would make me feel like a guinea pig. 50-50. I was concerned the information could be used against me. The government shouldn't have my samples. All 50-50, so they were concerned. But I'm curious about the influence of genes on health. 80%. Participating would make me feel like I were helping other veterans. 50%. My favorite question of all, I suspect the VA is doing this secretly already, which we were not. And then an issue about the validity of the research. I worry about the ability to recruit from all segments. I mean, that's a pretty insightful statement, and but overall they were interested in helping other veterans. Everybody wanted information about their health. This is a main driver. I'll tell you right now, we do not use a CLIA pipeline, and we are not releasing information. And that's an issue. I hope we can address that in the future. Remember, this was almost 10 years ago when we were designing this, and we're interested for the second $500,000 in doing it. People wanted some money, but they were very interested in having their privacy protected as well. Over 90% thought that was critical. So people are concerned with the privacy. So those were the things we took home. With whom should we share this information? 80% plus said U.S. academic medical centers. Okay. Government health organizations, we didn't stipulate that, but DOD, CDC, ARC. U.S. pharmaceutical companies, much more 50-50. And you get to foreign places, it's less than half. So I think we tend to be guided by this. So 83% said it would be done, 71% said they would participate. As is always, 71% are not actually participating. We're getting, on a good day, 60%, but over half are actually participating when they're approached with a vacutainer tube. And there were the usual covariants that you would expect with willingness to participate. So this became, after a year of huddling down in the basement of what was then the Armed Forces Institute of Pathology. Cell phones and beepers don't work down there. And so we would go down there for meetings to design what became the MVP. And we set out to collect a cohort of 1 million people, and just back of the envelope calculations looking at disease prevalence, disease heterogeneity, what do we need? We figured we could at least replicate a lot of the studies that we were most interested in, if that was the standard. We wanted to do it in 5 years. Obviously, we wouldn't. We're giving out a questionnaire, 2 vacutainers. We're saving plasma, buffy coat, as well as DNA, and the samples are de-identified. So the recruitment is really all comers. There are 0 exclusion criteria. The one inclusion criteria is you have to get your medical care at the VA at least at some point, because we phenotype through the EMR. Right now, everything is shipped to Boston. Some of you may know that there's a building going in across the street that will be, I think, the largest biobank in the world. Will have storage for 20 million specimens. Should be very nice. And then we're genotyping everybody up front. We decided to go ahead and do that en masse. And the whole mindset of this is that we're not going to, I won't say we will never send Will Dere 100 samples. But the idea is we'll process the 100 samples as part of a contract for processing 100,000 samples, so we use the benefit of scale, and then keep the data. Will can get the data, but so can other people. And so this is, and so it's really a centralized program. Here are the enrollment sites. You can see Salt Lake City is one. And as of, so this is a slide that I found that had been presented for the GEMPAC. So we've mailed out 3.7 million invitations. We've actually gotten consent completed on 520,000 at that point. And the hard part is the questionnaires. So we've had 600,000 questionnaires already sent in, even without the consents. So they're just filling them out and sending them in. They're great. We've completed over 200,000 genotypes. We've got another 200,000 in the pipeline being cleaned, and those should be available roughly now. Our target date was December 1st for 400,000 being available. We're using an AFI chip for these, so this is getting a little wonky on the genetic side. I apologize to those of you that are not geneticists, but so it's an AFI chip, a 723K, and we've put on 100K with specific clinical significance, indels, 300K that's part of a multi-ethnic grid. Some of these I know are shared with Kaiser. So we worked with Neil, I think, in the old days, so that we'll be able to pool data more effectively. And we're a diverse cohort. Again, this is a little wonky. This is a principal components analysis. We have 9% African American, and we're actually seeking to enrich and targeting some groups, Asian Americans, Native Americans, and then the VA, most of you probably know, is enriched for men. And so we're actually trying to recruit about 15% women. That'll be 150,000. That's not going to be representative, but it's not a small number if you think about it. The computing platform that manages every bit of this is called Genesis, and it stands for something. There's the corporate data warehouse is down in Austin, and it's primarily administrative data. Vinci, which is the Veterans Informatics and Computing Infrastructure, was really developed here in Salt Lake City. The hardware is in Austin, but it brings in all of the rich textual information and information that's not part of the core system. And I became aware in doing this of how many different data trails there were, the EEG reports, the radiology reports from the PAC system, respiratory notes. Those are all is all in separate systems. And so Vinci is sucking all of those in a high-performance computing environment that can do natural language processing. The data architecture structure is really, there's the survey data, so there's a 20-page survey. We do have permission to recontact. We haven't gone out to do that yet, except to pump out updates. There's the clinical data from Vinci. We have the death index and DoD data, and then of course the molecular data. And these all go behind this wall, and then we have governance as to who can get access, and we're just opening up access now. This summer, just as we passed 500,000, and President Obama got to announce it the same day, which was very cool, we got an agreement with DOE. And so even though we just bought some brand new computers, installed them in Pennsylvania, got them all shiny, plugged them in, it was all great, and they're just not going to be able to handle it. This is really big data. And so you say, well, what has extra CPU time? It's the DOE that does. And so the data are, as we speak, at the Oak Ridge National Lab. So the plan is Argonne, Los Alamos and Lawrence Livermore are all going to be participating. We're actually building a bridge between the VA system and the National Labs system. To do the first load will take almost a day to get the first pipe of data over there, but the incremental updates should be slow. So that's the slowest link down here in the corner. That little bit that we have to do is only a gigabit. So the current status, almost over 200,000 genotypes are ready, 200,000 more are ready to go, imputation on that is in progress, and we really look at an on-demand model. I'm doing okay. It should be okay. So now we're starting the science. What science is being done? This is the alpha test. We're mapping my dyslexia and typing that slide. No, this is, the first test was really approved at the same time the MVP was approved, and the MVP is the control cohort for the trial. And so we're carefully phenotyping 9,000 people with bipolar disorder and schizophrenia each, so 18,000 total. We've got over 9,000 already enrolled, and we're then using 18,000 of the MVP as a control, or maybe 36. We're generating exome sequencing data as part of that one. The second alpha test was the second one approved. It's wholly embedded in the MVP. The phenotyping is all done from the medical record, and it's on PTSD. So everything is going on is an intra MVP project. Just a year ago, they awarded the first 5 beta test projects. They were demanding that they be consortia, and so these are just now getting access. I think 3 of these, as of about a month ago, had access to the back end of the Genesis platform in Pennsylvania on cardiovascular disease, kidney disease, macular degeneration, complex metabolic portfolios, and polysubstance abuse. Phenotyping, for those of you that have done genetic research or have thought about it, phenotyping is a lot harder than genotyping. It's a lot harder. And so there was a phenotyping workshop a couple of months ago, how to get this out of the medical record. You know that in NHGRI they've had the eMERGE consortium, the electronic medical records for genetics, and something is a big deal, how to get these phenotyping out. So this was actually chaired by Matt Seymour, who's here at the University of Utah. And so we're really working on the phenotyping side of the platform. The future, we're anticipating an RFA probably coming out this spring for more data access. We'll have, I think, opportunities through this, through a new Department of Defense collaboration. We've partnered as of a few months ago with the PMI, so anybody who comes to the MVP will be offered enrollment in the All of Us consortium, and we're also going to be recruiting their spouses, which we can't do now. So we're going to be helping with that. The DOE agreement should help as well, and then pilots. I just want to close with a couple of other things we're doing. The VA started the Cooperative Studies Program way back in the 1940s. The first one was multi-drug treatment of TB. It was the first multi-site randomized double-blind clinical study in existence at that point. Right now we have the largest interventional clinical trial in the world running, over 500,000 enrolled in the CONFIRM trial, randomizing people to FIT testing annually or colonoscopy. It was a VA cooperative study that blessed you with colonoscopy at 10 years if you're if you have nothing on the first one, and 5 years if and that whole risk tree was all VA data. So there's a lot of things, aspirin for MIs. So we have a rich history of doing clinical trials and a clinical trial infrastructure. So using this, we've just funded the PRIME study. I think Salt Lake will be one of the sites. I think we get the money starting in January. It's a pharmacogenomic study for treatment of depression. Most of the pharmacogenomics are for oncology, and the black label boxes for do not use, a back of the urine, the right HLA type. There's precious little that really has shown clinical utility in pharmacogenomics in the general population. And so we're coming out with a clinical utility study. It's a mixed method study with delayed enrollment that's going to look at, on one arm, that's going to look at an outcome of improvement of the patient, but also how the physicians deal with that data and enroll it into their workflow, which is, I think, one of the things we need to approach. And then there's more big data. Somebody earlier today mentioned the Precision Oncology Project, the POP project. We're trying to sequence 10,000 solid tumors this year with a panel of 130 genes. We've partnered with IBM Watson Health to annotate those. Clinically, the annotation is done by N of 1, and then we're getting a second annotation by IBM Watson. We're standing up molecular tumor boards. We have an agreement now with the NCI, and we're going to try to stand up an enrollment so that any veteran can be enrolled. And on our aspirational goal, the VA is well known as a regulatory-rich environment, and we have some self-inflicted wounds. We often make regulations that are very difficult to follow. And the goal is to have, is to be able to enroll a person from diagnosis to enrollment in 72 hours. We can push 7 days. I think that'd be good, but we're really trying, and that's going to change a lot of VA regulations. REPOP is that Research for Proteomics, the Apollo Applied Proteogenomics, which is a DoD-NCI collaboration. We're using a BD STEP training program for teaching people how to use BD STEP, and then we have moonshot collaborations. We just got $50 million from the Prostate Cancer Foundation. And this is why it's good to work at the VA. And it's my last slide over there. The veterans are tremendously willing to enroll, and it's pretty easy for us. The model in the VA is that the researchers are clinicians, and they're expected to be on the wards in the VA. And it's pretty easy to take that out. I think we'll always remember is ongoing times at the VA. You know, he's gone back there because it's so good. So thank you very much.
So we'll hold the questions till after Dr. Schaefer's done.
Well, I wanted to thank you, Jim, for inviting me and the other members of the symposium organizing group. It's really a pleasure to be here today, and I found the morning sessions to be really enlightening and also inspiring, and so I very much appreciate being included. My plan today is really to talk a little bit about the history and development of our research cohort, and then I wanted to, if there's time, to give you a little bit of a sense of at least two examples of things that we're actually doing now with this research cohort that are precision medicine, or at least directed to research in service of precision medicine. And then talk about some of the implications of the cohort that we've developed, lessons learned, if you will, for precision medicine research going forward. So my colleagues and I at Kaiser Permanente Division of Research, and also Neil Risch, importantly, at UCSF, started working together on developing a resource for studies on genetic and environmental influences on health, disease, and aging back in 2000. We were just beginning to think about what might we do with these new developing genetic technologies to advance research, and we were approached by the SNP Consortium, who was interested in, they knew we had a large population, and they were interested in developing a database of human genetic variation. And so we said, well, that would be a very useful database, but it would be so much more useful if, in fact, it was linked to rich source of clinical information like electronic medical records and behavioral and demographic data from surveys and other data, genetic data derived, of course, from biospecimens, and environmental exposure data as well. And if we could bring all these together, it would make a very powerful resource for research. And so the SNP consortium actually said, oh, that's quite a bit beyond what we have in mind. We're pre-competitive. That sounds like the next stage. And so we spent another, I think, 5 years. We had we drew up a budget and sent out requests to foundations that had very large endowments. We'd figured out that it would cost us about $60 million to build what we had in mind. We originally conceived it as a resource of 500,000 adult members of Kaiser Permanente in Northern California. And we waited for the checks to come back by return mail. And it took about 5 years before we finally got a bite, and it wasn't for $60 million. It was quite a bit smaller from a local foundation for about $3.5 million, but it did get us started. So we knew we had a good basis in Kaiser Permanente in Northern California for making such a resource, and I talk about this a little bit because I think it does have implications for where you locate these resources, how you go about building them. Kaiser Permanente, the Northern California region, is an integrated health services delivery organization. Current membership is about 3.7 million people, although we have records and data on over 12 million. 30% of the population in a 22-county area of Northern California are members of Kaiser Permanente. It includes both rural and urban areas. The health service delivery is comprehensive, although no dental. It is generally representative of the population, although less representation at extremes of income, and has good representation of the major race ethnicity groups, reflecting the diversity of the population in that area. The membership is very stable. We have about 70% retention over a 10-year period, which includes mortality. So that's an important consideration if you're building a research cohort and you want to be able to follow them up and enrich the cohort as it ages and undergoes health status changes. So this is basically how we went about it as we gained additional funding from various foundations. We started with a mailed survey to virtually all our adult members in 2007. About 430,000 people eventually completed the survey. We then went back to survey respondents and asked them to give us a broad written consent and then provide a saliva sample using mailed saliva kits through the mail. And we then transitioned to actually approaching not just survey respondents, but all the members of adult members of Kaiser Permanente in Northern California. We integrated our specimen collection with our clinical infrastructure so that we're now able to place a research blood draw order for individuals who had consented to participate and be able to collect that and courier it to a dedicated biorepository as a part of the sort of using the clinical infrastructure in a way that was cost efficient. And now, in just the last couple of years, Kaiser Permanente has decided to expand this cohort to include Kaiser Permanente members from all the regions across the country. And we now consent electronically and contact people largely electronically. It turns out that things have actually changed since we were doing things largely by mail. Now everybody ignores letters, nobody gets mail anymore apparently, and so it's mostly done by email, and we're using some other recruitment methods as well. So that's added about another 50,000 individuals in basically the last year to the cohort of 200,000, a little more than 200,000 that we had established in Northern California. So obviously a huge important component of creating a research resource like this are the existence of comprehensive electronic medical records. We have had comprehensive electronic databases since 1995 that included virtually all the domains of medical care and information about medical care. Everything but physician notes actually was electronic, and then developed a fully integrated electronic health record in 2006 that really covers all domains, all encounter types. Now, we've heard a lot about what it takes, the complexity of dealing with these very large bioinformatics files that are generated as a part of generating genetic information on large numbers of people, but dealing with the sort of echoing Larry, the complex, multidimensional, longitudinal aspects of using electronic health records, which are designed to support care, to make those research ready and be able to really access that information in a way that makes it useful for research, is a huge amount of work and something that we have put a lot of resources toward. We have developed a virtual data warehouse production research database that's refreshed daily, but there's lots of other work to do to really get standardized, organized, and integrated information for studying different phenotypes out of such a comprehensive amount of data, granular data. In terms of environmental influences, well, there are questions in the survey, and there are environmental exposures that can be measured in the serum samples, for example, that we obtain. But I think another important source of environmental information that's actually at a different level of analysis comes from a geographic information system that allows for sort of developing data that's linked to geographic coordinates, such as U.S. Census block groups, for example, for the study of local environmental conditions and neighborhood characteristics. So we're very lucky in California that we actually have large publicly maintained databases on things like pesticide exposure, water quality, air quality, and these databases go back actually decades, in fact. So this information can, in fact, then be linked for participants in this cohort to longitudinal residential information, for example, and give you information about environmental exposures. And we also developed a social and built environment database as well. So very importantly, I usually have one of my colleagues talking about our stakeholder engagement and bioethics input, but I wanted to tell you at least a little bit about this. So this is a little bit sketchy, but from the very beginning, we gave consideration to ethical, legal, social implications. I think in part due to the fact that we are creating this within the context of a healthcare provider, and one that frankly was a little bit nervous about our going to all our adult members and inviting them to be part of something that, recall, this is like 2005, 2007, still pretty novel idea, and some concern about whether members, how they would respond, how they would react to this. We wanted to make sure to do it right because we have a long-term relationship with our members, and that's multifaceted. So we created a community advisory board in 2007, and that's actually ongoing up through the present, with 3-year terms, about 20 members, quite diverse communities represented, as you would imagine, in Northern California. Also a bioethics advisory panel since about 2008. With external experts representing a variety of different areas of expertise. We sort of cross-fertilized these panels, had members of the scientific advisory panel and the community advisory panel meet with the bioethics advisory panel and vice versa, so they all knew what each other were talking about and thinking about. And I have to say that the Community Advisory Board was actually very instrumental, for example, in determining that we really needed to maintain our trust with our participants and with our members by doing an opt-in reconsent when we were going to do a large deposition of data into dbGaP. We had a broad informed consent that included sharing of data with external investigators, but we felt it didn't explicitly call out dbGaP or federally maintained databases. So it turned out to be a very good decision. We got over an 80% response rate in terms of people reconsenting, actually sending back a new signed consent form. So then in 2009, by benefit of the ARRA funding, we were enabled to genotype over 100,000 of the participants in the cohort. It was titled A Resource for Genetic Epidemiology Research in Adult Health and Aging. We call it the Dere cohort. This was a collaboration with the UCSF Institute for Human Genetics. We custom designed ancestry-specific genome-wide SNP arrays and used these to genotype over 110,000 participants with about 675,000 to 890,000 markers, depending on the array. Short amount of time, because that's what the funding called for, using the Affymetrix Axiom platform, which is the same platform as the VA has now used, and also the UK Biobank facilitating harmonization of that data. And then imputation of data to 1,000 genomes, and we also measured telomere length on the same 110,000 individuals. And then came the work, of course, of finding ways to really easily link these data with extracted data from the electronic medical record, geographic information system databases, and other databases. So this will just give you a little bit of a sense of what the cohort looks like. Majority women, and you can see that over half of the people in the cohort are 60 to 79 years old, which is great for studying chronic diseases. The population of Kaiser is actually a lot more proportional with respect to age, and it just turns out that 18 to 39-year-olds are actually difficult to enroll in this sort of research, which I think is a consideration going forward. You can see, too, that while we do have representation of the different race ethnicity groups, that, too, is not proportional to what it is in our population and our membership. And while you don't need perfect representation or proportional representation to have a robust cohort, we would like to do a lot better than this. We would really like to be able to include much larger numbers of minority participants, and that's a key focus now of the expansion to other regions. And you can also see it overrepresents, to some extent, individuals with more education, which is typical of all research, I think, to a certain extent, but is something to consider. And then this is an old slide. This was probably done about 3 years ago, and so actually the numbers of individuals represented here, particularly in cases that have occurred after specimen collection, is low. But the primary thing I wanted you to see from this is that, of course, there's good representation of common diseases in a cohort of 100,000 people. But there's, a lot poorer representation of rare diseases. And in particular, I think this has relevance for recruitment, for example, for precision medicine, or the All of Us study, is that diseases like schizophrenia, for example, are not represented in proportion to the numbers that we would expect in this size population and with this age and gender distribution. So there are health conditions for which that are underrepresented in research and for which special efforts would have to be made. If you want to include those diseases in what your research cohort is capable of addressing, you'd have to make probably special efforts to overcome the issues that keep people with psychiatric disorders, substance use disorders, and so forth from volunteering as frequently as others. So getting the signal, I actually have to stop without giving you a little bit of the research focus here, which is a shame. Actually, maybe I'll wait then to talk about implications and hope those come up as a consequence of the questions when we get them. Thank you.
So we have about 20 minutes for discussions, and I'll open with the first question. Dr. Schaefer, why don't you tell us about one of your examples?
Well, that's so nice. I was going to tell you about a genome-wide association study of bipolar disorder that we did. So this came about the same time as the. Actually, it'd be great if I could show the. So, let's see here. So we conducted this genome-wide association study of individuals with bipolar I disorder. It's about 5,300 people. At the time, that was a very large study. At the time it was funded. And we thought that this was probably the largest study of bipolar disorder at that time. Thank you. And we had controls from the Dere cohort. However, it turns out that this is an example where you can see that actually 5,300 people with bipolar disorder is really not enough to learn much about the genetic architecture of bipolar disorder. So in fact, what we've learned with psychiatric disorders, but other disorders as well, is size is very important to discovery. So meanwhile, there was a large genome-wide association study done by the Psychiatric Genetics Consortium, putting together a lot of different studies, doing a meta-analysis of schizophrenia that identified 128 genetic risk variants for schizophrenia. And so we wanted to ask the question of, can genetic risk variants for schizophrenia explain some of the clinical features that are overlapping with bipolar disorder? And so I think of this as precision medicine because in the 2011 Institute of Medicine report, they talked about that one of the things that we needed was a new taxonomy of disease. So precision medicine and precision prevention isn't just about how we treat disorders, but also our taxonomy or our finding the basic underlying reasons for disease and potentially reclassifying or thinking about how we our sort of nosological categories somewhat differently. So there's reason, obviously, to there are certain shared clinical features for schizophrenia and bipolar disorder, such as psychotic symptoms and suicidality. We know they share genetic liability from family studies, from a variety of other studies that have been done. And so, let's see if I can make this work. So what I wanted you to see was that, in fact, using for the individuals with bipolar disorder, scoring them, developing a genetic risk score for each of these individuals for risk of schizophrenia, genetic risk of schizophrenia, basically it's that schizophrenia genetic risk score is extremely strongly associated with psychotic symptoms in individuals with bipolar disorder. And similarly, psychiatric hospitalization for bipolar disorder also very strongly associated. That's an indicator for severity. So essentially, you're using the significance of this is you're using now a biological indicator to actually dissect this phenotype of bipolar disorder and begin to understand what some of the specific phenotypes within bipolar disorder, what the explanation, some of the genetic explanation for those might be.
Thank you. Questions? Right here.
Hi, I have a comment for Dr. Meyer and then a question for both speakers. My comment is I worked with Jonathan Simons in the mid-2000s at Emory University Winship Cancer Institute. He's now the president, has been the president of the Prostate Cancer Foundation. So intriguing that you received a $50 million grant. He was the first recipient of a grant back in the 90s as a young researcher of $50,000. And the reason Michael Milken created the Prostate Cancer Foundation was to try to help find answers. And so he put all of his resources into that, and he got his grant in a month because they couldn't get money from the government. It took so long to get an NIH grant. So intriguing that it's come full circle, in essence.
Full circle. And so Michael Milken was there on the Tuesday event, and he also had an editorial in the Wall Street Journal that day, I think on either Monday or Tuesday. For those of you that don't routinely read that, it's available online and very nice.
I'll check that out. Thank you so much. And then for you both, I don't believe I noticed, but I'm curious about the governance, regulatory oversight of your projects, and then ongoing monitoring, internal monitoring for verification of data. Is there an element of that occurring around this, an institutional board of governance of some kind?
So I cut a lot of slides. We have a complex web of oversight, internal and external advisory boards, project-specific boards, data governance, the governance of the molecular pipeline. It's I won't say it's extreme, but I think it's tight, and so and then getting access to the data are all well controlled. So I think we were really well informed by the veterans initially. We continue to have veteran engagement at all points because they're our bosses. Well, Congress is our real bosses, I suppose, because they pay us. But then the veterans are the people that we serve. And so we went initially for a full consent. So we don't use, it's not an opt-out, it's an opt-in with a consent. And so yeah, we do have governance at all levels.
So actually, we have a relatively small and discrete governance and oversight by, in addition to the boards that I mentioned, there's a relatively small internal group of health plan leadership that basically is the governance for this research program.
Thank you. In front.
Thank you. Thank you. I have a question to Catherine. I'm wondering if you can say a little bit more about the study with people with schizophrenia and bipolar. In particular, I'm interested in learning more about how you incorporate into the study environmental factors and sort of the other parts that comprise precision medicine besides the genetics. And of course, the one other question is, how do you keep participants with these conditions engaged over a long time?
So I'll just say something about the second part first, because I think actually that's one advantage we have with this research program occurring within a healthcare delivery organization, is that actually does, they're by virtue of being members of this health plan, they continue. We send them try to send them regular newsletters, keep information posted about studies that are ongoing, but we really do very little to maintain their engagement. We haven't found that that's particularly a problem, in part because I think of their ongoing membership and the fact that that maintains contacts for them and so forth. This particular study is not a good example of bringing in environmental factors, I think. This is really focused largely on the genetic architecture of bipolar disorder, for example. We did do an interview which includes aspects of lifestyle and some history, and incorporate that as part of the model of factors that influence development of bipolar disorder. But this is not, for example, this is not a good example of using the geographic information system to.
Could you maybe say a little bit about both the retention and the environmental data for the Million Vet program?
So environmental data overall, I think, is a very hard nut. So we do have an entry-level questionnaire, both a brief one and then a much more extensive one. You saw that they are being filled out, and that involves everything: burn pits and Agent Orange. So we have exposures on that realm, food frequency questionnaires, other environmental data that picks up on. So we do have that. Now that's going to be incorporated into the analytical pipelines of individual studies, not at this point in any broad category. We've just gotten clearance to start reaching out electronically. So to follow up on the previous question, we do send out newsletters, but yeah, the veterans do come back in any case, and I don't think we lack for engagement. It remains to be seen how engaged they'll be with different forms of electronic communication and their willingness to fill out quick things on an iPhone app, which I think will be probably the best way of doing it, and getting that information.
Thanks. Yeah, we have time for 1 or 2 more questions in the back. Yeah, right back there, Mike.
Hi, this question is for Dr. Schaefer. I was wondering if you had any theories as to why it's harder to recruit the 18 to 39-year-old population?
Well, I guess conventional wisdom is they think they're immortal, and this sort of thing is largely. But I think, particularly young men seem to have better things to do than complete questionnaires and spit into kits. So I think it's just we haven't found. We're looking now, actually, sort of through focus groups, exploring different ways to reach young men, and particularly young men of color. And I think appealing actually turns out to altruism, to a sense of contributing to the community, is quite powerful. That by virtue of their health and youth, they have a lot to give, and that may not be obvious, the sort of standard way we talk about recruiting people into health studies.
I can address that as well. We've seen exactly the same thing. We have a tremendous overrepresentation of the Vietnam era over the Iraq and Afghanistan era, and I think one element is the trust that they have in the system. And another is the way that we engage them, because our initial contact is by a letter. Great. right? And as I said, we're just getting permission to use email, to use electronic consent. And that'll also mean that we get truly nationwide enrollment. So that'll open up a lot. But I think we have to use different means of communication with 25-year-olds than we do with 70-year-olds.
Another question? Yep, right.
I'm a retired veteran, and when I drive around this city, as opposed to probably every city, and I know the veterans are the largest group of homeless people, what's being done about that? I mean, well, there's a guy on every corner that's got his cardboard that says, "I'm a veteran, you know, anything will help," or whatever. But the bottom line is there any way to hook them people and bring them in? I mean, I'm sure some of them must need medical care.
Yeah, so enrolling homeless people in studies is one aspect. I think the VA has made a tremendous effort starting under Secretary Shinseki to address homelessness. The goal is zero homelessness. Officially, both Utah and Arizona have now hit zero homelessness. We have a policy here of housing first. When we find a veteran in Utah, they can get a clean sheet bed and a shower and a locking door that night. No questions asked. If they come in, we can find them. Not everybody that has a sign that says they're a veteran are. Not everybody is willing to come in either. Now, in other communities nationwide, we know we've reduced homelessness over 40%, almost 50% nationwide, and that's good, but it's not good enough. And you know, clearly the U.S. has an issue with homelessness as well. Housing costs, social factors, health insurance. I could go on. I suspect we'd agree on a lot, but I can go back to the study that we're doing that was informed by the study that Kaiser did on mental illness. And so one of the things that we're looking at is functional capacity in mental illness as part of the phenotype. Because some people with mental illness can keep their home, but mental illness is clearly a reason for being homeless. And so we're addressing polysubstance abuse and mental illness, and that's one way that we can approach these much larger societal problems.
I'd like to generalize the gentleman's question. The individual who's homeless doesn't need his or her genotype sequence to address the problem that they're facing, right? And one concern that's often raised about precision medicine interpreted as genomic medicine is it's not necessarily going to address the real widespread health problems that many people in the United States or the world face. And so I wonder, do you have any examples of how your programs are perhaps addressing an issue like health disparities among the haves and the have-nots in the country, where it is helping that population? It's not just about finding increasingly expensive cures for fewer and fewer people.
So yeah, I mean, we have ongoing research. We talked about the Prostate Cancer Foundation. Blacks have a higher incidence of prostate cancer and a higher death rate from prostate cancer. Black women have a higher death rate for breast cancer and have a higher and are found at more advanced degrees. And so this is a complex interaction of access to health care along with genetic factors. So clearly, doing the genotype of somebody sitting on a corner outside of Home Depot looking for a job or a handout, whatever it may be, getting their genotype won't help them as a one-off. But I think looking at different genetic factors, people with, blacks have different responses to drugs in hypertension than do white Americans, and learning how to better treat people of different ethnic backgrounds, whether it's Native Americans, Asian Americans, African Americans, I think can improve health as on population basis. So one thing is the N of one kind of studies, but the other thing that genomics can do is let us approach populations.
Thanks. Dr. Schaefer, did you want to add something?
Well, that's a really excellent answer and hard to follow, Larry. So I think we're, for one thing, very focused on the issue that we don't increase disparities through the development of treatments, approaches, and so forth that essentially further disadvantage people who have historically been, at a disadvantage. But we do have active disparities research also going on. I think one of the things that our community advisory board told us very strongly is they were concerned that the sort of whiz-bang aspect of genetic technology would sort of have people lose sight of the fact that disease obviously is not all genetic, or even necessarily mainly genetic, and that we do have to pay attention to the environment and the context and the the social determinants of health as well. So I think we're trying to do that in our research program, but I'm not sure. I wouldn't claim we're 100% successful. So.
Thank you. We've reached the hour mark, so please join me in thanking our speakers. I think that's yours. I think you do, yeah.
And we'll just take 5 minutes in between the two sessions, so please stick around. Okay. So here we go. Hold on. Then we'll just minimize hers. All right. Weird. Close that one.
That doesn't say you have to put it in a public access bulletin. It's on the DOE server, and you have to get your training and get approved to come in.
There it is.
Yeah, we'll have. Okay.
All right. You want to just take it from here?
Oh, yeah.
Okay.
I'll introduce myself.
I'll introduce yourself. Okay. The pressure is off.
It will only take 20 minutes or so.
I've been rehearsing all of your illustrious titles.
Right.
You have a lot of titles, actually, Jeff, when you look at the list of the...
The next presentation will be up. Okay, I'll just put it up now then.
Yeah. Go ahead. Perfect.
We're just going to sit, right?
Yeah.
We can be cool.
Are you going to do that?
Well, do you have a preference, Jeff?
No, I'm here. Whatever you think is good.
Yeah.
How do you pronounce your last name? Kaiser.
Nope, fag. Oh. Everyone doesn't, or anyone thinks it would not be that. Yeah. But it is. So apparently these don't go off. Have to watch what we say. We were going to go first, right? We did it. Or we tried. Yeah. Aww, that's really cool. It's a surprise. Yeah. Fun to do with you. It's weird to like stand up. Our own colleagues left right after the last panel, and I do really fun. Like a bunch of people from Huntsville, and I'm sitting right here. I know you are not going back to the so you're going home. Worst thing about training at Harvard, where everybody pretended that they didn't do anything about it. I will everybody like when we went to Hawaii. Yeah. I put it away going like I'm in Hawaii. Period. With my husband. Period. With no children. None of my 3 children. Period. Don't even think about getting an email response for 10 days. I have already crafted my out of the office message, which is I am in Bora Bora, which is where I'm going to be a week and a half from now. And I'm not checking my phone. So don't even bother. Yeah. My dog is kind of weird. Oh, really?
Well, in a way, she may be better than Jeff Miller, which was the right time.
We're sitting here because we're a woman enough to do that. Yeah. Thank you. Yeah. Happy to.
That's what his.
Shut up. I always make those times. I've done stuff from the system. Shut up. Seriously, John. People are done with you. Just sit up. Angie will like poke me at that point and be like, seriously, stop. Seriously, it's my turn. Yes. I'll just start talking. Exactly. Grab the mic away. We have a very serious topic. I know. Yeah, I know. We got to get in like professional mode here. Yeah. That's okay. That's okay.
Yeah. Are you going to come to this evening?
I'm not. Great. Nice to meet you in person. Yeah. I feel like we might have met this sometime, but yeah.
I'm horrible with names.
All right, we're going to get started with our next session. I'm Jeff Botkin, Associate VP for Research here at the U, and head of the University of Utah Center for Excellence in Ethical, Legal, Social Implications of Genetic Research. And so my pleasure to lead or moderate this session that we're calling Engaging the Public in the Program. This whole notion of public engagement really is relatively new in the last 10 or 15 years or so. And I think the very presence of this segment of the program on this sort of platform is illustrative of that, as are the principles that Dr. Austin had laid out for the what is now called the All of Us program. Almost every one of those had something to do with public engagement and transparency. I had pulled off a couple of principles from the Precision Medicine Initiative back when it was called that, and they had 6 principles they articulated, and 3 of those spoke quite directly to this notion of engagement. One was building trust and accountability through transparency. Second was respecting participant preferences. And a third, empowering participants through access to information. So again, this whole notion of access to information is really so new in this particular domain. And it's by no means a simple set of problems to address. So why are we addressing these issues? I think the simple answer is we've politely phrased, not done a very good job with engagement historically. And that spans from a whole segment of research, some of which we've heard about today, which is data analysis and access to tissue samples, which involves no engagement whatsoever. Now, our work and many others who have engaged the public around these sorts of projects find that the public is supportive when they understand what's going on, or at least most of the public. But there's a solid segment of folks who very much object to this type of work. And those sorts of objections pose a threat to the enterprise that we're talking about and the trust that the public has in our institutions. The other type of problem that we've had traditionally is so-called helicopter research, where investigators will either come into a community or come into a disease population group, and they're your best buddy for a period of time until all the data is collected, and then they're gone and no further sign of them. So this notion of engagement we're defining, I think in this context, as a full spectrum of relationships, from engaging communities or engaging individuals with disease entities to try to craft research projects that meet their needs, to the informed consent process and the education process, to providing information through the course of the research, and at the end of the research, things like study results, whether that's individual results or maybe the general results of the study itself. And again, as Dr. Austin had emphasized, people do want this. They expect it, and they're rather dissatisfied when we don't follow through. So we're privileged today to have two just outstanding scholars in this domain who we're so privileged to have at the University of Utah. And I think your program may have them in a different order than what we've decided to do. Our first speaker is going to be Dr. Kimberly Caffings, who's professor in the Department of Communication and an investigator in cancer control and population sciences at the Huntsman Cancer Institute. Dr. Caffings is a member of the Utah Center for Excellence in LC Research and Family and Health Research Utah. She has had quite a bit of work that's focused on genomic literacy in particular, and in developing methods for returning genomic sequencing being one relatively recent example of the challenges that have emerged with the technology, and really engaging community participants also in helping to understand biobanking enterprise. Dr. Angela Fagerlund, or Angie, is just joined us in the last year or so. She is our inaugural chair of population health sciences here at the university. And her research is broadly based in provider-patient communication. Lots of work in developing tools to try to assess and enhance communication between investigators and research participants and patients and clinicians. Examples include a plain language decision aid for veterans diagnosed with prostate cancer, decision aids for breast cancer patients, and also of note, a role in doctor-patient communication and pain-related racial disparities. So they are both well situated to help us think about some of the challenges and opportunities we have to do a better job with engagement in the conduct of research. So Dr. Caffings.
Great. Thanks so much, Jeff, and thank you for the opportunity to talk this afternoon. Are you hearing an echo? If I'm too echoey, just let me know. All right. I will lean. Yes. Have not mastered the mic yet. So what I'd like to do is talk about one particular issue that I think is critical in thinking about patient engagement in biobanks or more generally in these sort of large-scale population cohorts that we've been talking about today, and that is health literacy, which is an area that I have worked in for about 15 years or so. So, of course, I think I don't need to tell this audience, but will anyway, that research with biospecimens raises, of course, important issues with informed consent. From a communication standpoint, one of the things that's challenging and very interesting, of course, is that there's a lot of information that you could potentially convey. So this is just one list of, actually a part of a list of the types of information that should be included in a consent form for a biobank that collects biospecimens. So secondary uses of biospecimens, indefinite storage of the specimens, ongoing medical record access, large-scale data sharing, privacy and confidentiality, access to research results, and ability to discontinue participation. As you can imagine, these are concepts that are not necessarily familiar to people, and they use a lot of technical and often quite legalistic jargon. But it's important that people actually understand the key elements of a consent in order to make an informed choice about participating in studies. The bad news, and was alluded to in the prior session, is that we know that there are major gaps in what people understand even after they complete an informed consent process. So people sign the form, but they don't necessarily understand all of those key elements that I just read off for you. And in thinking about this, the Institute of Medicine and other leading researchers have identified health literacy as a factor that seems to be incredibly important that affects whether people understand the information in an informed consent process, as well as whether they can make an informed choice. And this is just the cover from an Institute of Medicine workshop that happened last year on informed consent and health literacy, and I'll talk a little bit more about that at the end of my time. So first, just what is health literacy? Because I think most people who are unfamiliar with the term think of it as reading skills. But the way we think about it now more in the field is that it's a relatively complex set of communication skills that is really quite important in how people understand and use health information. So this is a definition. There are many different definitions. This is the most common one in the United States. It was adopted by the Institute of Medicine. So health literacy is the degree to which individuals have the capacity to obtain, process, and understand basic health information and services needed to make appropriate health decisions. So that's a lot more than reading. And I think the other thing the IOM did that was really quite useful is think about component domains of health literacy. So there is conceptual knowledge, which is the background knowledge that we often assume people have and that they need to understand our health messages. So even something as simple as it's important to get an annual mammogram assumes people understand the difference between a screening and a diagnostic procedure, for instance. There's also oral literacy, which is listening and speaking skills, print literacy, which is reading and writing skills, and I think what most people think of, as well as numeracy. And there's a good deal of research evidence now that print literacy and conceptual knowledge are incredibly important in how people understand information in the informed consent process. My personal opinion is that oral literacy is also quite important, particularly if there's a dialogue involved, but is quite understudied and I think would be really interesting to look at. So the best available data from national samples are from a large survey called the National Assessment of Adult Literacy, which the U.S. Department of Education does about every decade. This is from the last cycle. They haven't released completely the report from the most current one yet, but it seems to be very similar. So what this is showing you is that about 14% of U.S. adults fall into what's called the below basic health literacy group. These are people who are likely to struggle with literacy tasks on a day-to-day basis. The reading level of this group is more or less fourth grade of reading or less. There's an additional 22%, which is the white box, and the box I was just talking about was dark. I think it's coming out as sort of dark purple. 22% in the white box are people who generally have the types of literacy skills they need in order to function on a day-to-day basis, but they are likely to struggle in the healthcare setting. So you often hear in the health literacy literature that about 36% of U.S. adults have limited health literacy, and that's where this figure comes from. But I think it's really important to note with informed consent that it's really a proficient level of task, the highest level of health literacy task. And the diagonal box are the number of U.S. adults who have proficient levels of skills, and that's 12% of adults, which suggests that 88% of U.S. adults are going to struggle with reading informed consent forms and understanding the information. So this isn't a trivial problem that's affecting just a few people. This is a widespread problem in population-based research. And also, as we start to talk about recruitment of more underserved communities, it's also critical to talk about inequities in health literacy in the United States because they are quite substantial. So there is It's not even so much a gradient as a threshold of a relationship between age and limited health literacy. So as you can see, if you look at the dark purple and the white boxes together, about 3/4 of adults aged 65 or older are considered to have limited health literacy. So that is quite a chunk. Sorry, it's about 2/3 of US adults. But in comparison, only about 25 to 30% of younger adults, so in the age 19 to 24 grade, for instance, 31% of adults are considered to have limited health literacy. We don't know why this age threshold exists, but it is consistently shown across studies. There are also important differences by race and ethnicity in limited health literacy. So I should say before I say these results that the NAL is only conducted in English. So some of what you're seeing is the fact that these people may be literate in other languages. But I think the data still shows that there are substantial inequities. So individuals who identify as Black or African American, Hispanic, or American Indian or Native American or Alaska Native are more likely to have limited health literacy than individuals who identify as white or Asian Pacific Islander. And then I think, as you might expect, there's a very strong relationship between educational attainment and limited health literacy. So a very large proportion of individuals who did not complete high school have limited health literacy, and only 1% of that group is considered to have proficient levels of health literacy. In comparison, people with a graduate degree, there are still 13% of people with a graduate degree who are considered to have limited health literacy. This may be because they did their graduate degree in another language. That's hard to disentangle from this data. But still only 33% of people with a graduate degree have proficient levels of skills. And so just being educated is not necessarily going to mean that you understand the consent process fully. So what do we do about this? I'm not trying to say that this is impossible, that we can't consent people in a meaningful way. But the way we think about this in health literacy is to bring down the health literacy demands of our materials. So how do we lower the literacy demands of consent in this context so that it is a better match for the skills that people have? Because if you manage to close this gap between demands and skills, that leads generally to better comprehension. So I'll just show a little bit of data from the print literacy and conceptual knowledge realms. So there are literally at this point hundreds, if not thousands, of studies that have shown that consent forms are just not a good match for what we know about the reading skills of most American adults. So we know that the average U.S. adult reads at about an 8th to 9th grade reading level. People tend to read about 3 to 4 grade levels lower than their highest educational attainment. So generally people on average have a high school diploma, but they read at about an 8th to 9th grade level. But there are a lot of studies that have shown that very few informed consent forms are written at less than a 10th grade level, although I think this has actually improved over time. But even still, many still demand college level reading skills. And this diagram, thank you, on the right shows on the right hand, the bars show the number of consent forms that surpass the target skills of the population, and on the left shows the consent forms that have a reading grade level that is lower than the target skills. So I think what you can see is the consent forms are almost always more difficult than the actual skill levels of their population. Another challenging thing is, of course, conceptual knowledge. Language is one thing, knowing the concepts is another. Informed consent documents and processes more generally contain many unfamiliar and technical concepts. So one study that I did at the Dana-Farber Cancer Institute in Boston looked at people's conceptual understanding, and these were all participants who were actual biobank participants, so had signed the form and hopefully had given informed consent. But one of the questions that was quite striking was we asked how they interpreted a line in the consent form that they could withdraw from the study at any time. And this is an answer from one of the participants who said, "What for? In formaldehyde on my little shelf at home? Oh, there's my cancer honey? No, I don't think so." And so this was actually a fairly typical response, and we disentangled what was going on. And I think it's a problem with the analogy to a bank, because withdrawing something from a bank means you get something, but also that they didn't associate the word withdrawal from stopping the study. And just also to raise the point that this was actually a college-level chemistry professor who said this. So it's not just that it's people with only low levels of education. So there are currently recommendations for simplifying consent forms. So for example, having a one-page informed consent form that they say is outlining important issues and risks in straightforward language. This is a lot easier said than done. It is actually quite difficult to write in straightforward or plain language. It's difficult to find synonyms or definitions for some of the types of concepts that we want to convey for biobanks, but also deciding on, if you only have 1 page or 2 pages, what is the most critical content to cover. And simplifying only words might be not enough. So we now have a number of randomized studies that have compared a simplified consent form to a standard consent form, and comprehension does not always increase when you lower the reading grade level alone. So we've moved on to more using clear communication principles, which are pretty well established at this point in the field of health literacy, but are starting to be applied to informed consent documents. So using common words, writing in first person and active voice, keeping your sentences short, only one main idea per paragraph, clear organization, descriptive headings, and adequate white space. All of this is just to say, make the information more relevant and salient, and also make it clear what you're saying and why you're saying it, and try to use as simple language as possible. This is, of course, again, easier said than done, but I think it gets a lot easier with practice. You probably can't see this, but this is the actual words, but this is to highlight there are a number of researchers around the country who are doing really excellent work in this area. So this was an informed consent document from Laura Besco, which used some of these principles. So clearly organizing the information, you can see that there's some highlighting to highlight key points, and really trying to make it more personalized and relevant for individuals. This is just 2 examples from one of my own studies to develop an informed consent form for a biobank for breast cancer research. This was a very humbling study. We did a lot of formative research, so we did interviews and focus groups with the target audience and showed them a whole series of prototypes to try to convey some of the more challenging ideas. So I think on the left is a pretty typical example of what the text looked like. I have to say we had a really excellent community advisory board who had a lot of opinions, anything from the type of font we were using to the exact blue. I never knew how many different types of blue there were until we started talking about this. But I wanted to highlight also the graphic on the right, which was something we spent a lot of time on. One of the concepts that was incredibly difficult is the idea of secondary and unplanned uses of biospecimens in research. And it was just very difficult to convey the idea that we don't know what we're going to do with them. We have general ideas, but we have lots of different possibilities. And so this particular graphic went through 7 different iterations with testing with focus groups, revising it, and actually these were all the different images that were suggested by our community participants. And in the end, I've been proud to say it actually conveyed in a randomized experiment, comprehension of this concept was very high. And the one point I also wanted to be sure to make is that comprehension was higher both among our participants with low literacy, but also our participants with high literacy. And they all preferred the simplified document compared to a standard document. So this is actually beneficial for not just people who have limited literacy skills, but participants more generally. So then just in my last point, I wanted to say that the other thing we are now thinking about is not the exclusive focus on the informed consent document itself that I think the field of health literacy has had, and start to think about the entire process. So the first 3 steps are really focused on knowing your target audience, knowing what information you want to convey, and being clear about what the key information is. The fourth step there is writing your simple document. You can't get away from that. It's important to use clear language. But then really emphasizing the dialogue as well. So what is the oral communication going on? Because somewhere around 15 to 20% of US adults don't actually read well enough to get much information out of written information. And so it's really important to be thinking as well about oral or verbal communication. Also considering alternative formats, video, multimedia, online formats. I think we have a tendency to default to the written form, but that's not necessarily useful, particularly for people with limited skills. Assessing comprehension. So there are techniques that I can talk about with any of you who are interested for both assessing comprehension as well as improving it. So things like teach back, for instance, and then having an ongoing evaluation. And I will skip over this for the minute just because I think I have gotten my stop a couple minutes ago. But I think that is just a general introduction to some of the issues with health literacy and informed consent, and I look forward to talking further.
That was great, Kim, and thanks for everybody for sticking around. While the slides are coming up, I'm going to start by saying that I'm going to connect this a lot to what came out in Dr. Ulrich's section, talking about how do you communicate this information, maybe the risks and benefits of taking a baby aspirin to prevent colorectal cancer, or some of the other examples that they were giving. But before I do that, I want to start by a little story. For reasons I won't bore you with, a couple years ago, my pediatrician suggested that I have my son get a genetic test. And when the pediatrician sat me down and gave me the results, there was a mutation that was, I don't know, missing or wrong. This is how I understand this. Was wrong. It was missing a little part or something like that. I'm very sophisticated here. And I go, oh, I'm sure there's always little mutations here and there. There's nothing to worry about. He says, no, I've actually never seen this before. I'm like, okay, great, thank you. And I'm like, so what does this mean? He's like, well, I don't really know, but I can get you set up with a genetic counselor in a couple of weeks. I said, well, I'm going to go, text my postdoc who's a geneticist, and I'll ask her. But what I'm telling you in the story is not to give you a war story about being a mom. I have a lot of those, but that's not the point. The point is I had a really good pediatrician who couldn't explain anything of the test results that he was giving me. I, as a Ph.D. who had actually already had a merit award from the VA studying risk communication and genetics, couldn't understand the information, and the only way I could get any information was by talking to a buddy. That is showing just the flaws that we have in understanding and communicating about genetic risk information to patients and their family members. So in here, and I was really hoping that this would show up better, but this is just a great example of a typical genetic test result. And you'll see over and over again, it says uncertain clinical significance, uncertain clinical significance. That's what they ended up saying about my kid, is of uncertain clinical significance. So what are we supposed to do with this as patients or family members? And what are providers? Should you be presenting this information to patients if we don't really know what it does, because we know it's going to worry them? I mean, I hear that things that says unclinical significance worries mothers, but maybe that's just me. So the question is, how do we present this information and what should we be presenting? Here's another example where, again, this is what is given to patients. This is given to providers who do not understand, oftentimes do not understand basic genetics. So again, it just is giving a lot of letters and not really explaining it in any kind of plain language so that a provider could sit down and talk with their patients, or that patients could understand when they take this home with them. So this leads to so many questions and so many problems. What do we do with the uncertain clinical significance? What do we present? What do we not present? Is it better to not tell patients this, or is it better to give this information? How can we improve providers' ability and self-efficacy for both referring people to genetic tests and communicating genetic test results? And then finally, how can we help patients be more understanding of this information and engage in genetic testing decisions, whether to be tested or what to do with the information after they've been having this information presented? So let me kind of give you some examples of some of the problems that we're seeing right now. First of all, in a great article that by White and colleagues, as well as some colleagues from here at Utah, really what is being found is that physicians are most often referring people to get genetic tests because a patient asks. A lot of times it's not because the physician thinks it's the best thing for the patient or there's good medical evidence, it's because the patient asks and they don't want to say no. That's not necessarily a good reason to ask people to undergo genetic testing. Second, there are disparities in who providers talk to about genetic testing results. My wonderful colleagues, Reshma Jagsi and Sarah Hawley and their colleagues from University of Michigan looked at breast cancer patients, about 1,500 different breast cancer patients who had recently been diagnosed with breast cancer. And the Latino and African American women who had been diagnosed with breast cancer were far less likely to be talked to or even asked or even given any information about genetic testing results. And furthermore, people who did not have a satisfying conversation with their physician about genetic testing had much more distress about their cancer, more distress about reoccurrence. So not only was there a disparity in getting tested and the information that you can gain from that, but also in the emotional and outputs of that, outcomes of that. And on the provider side, Birmingham and colleagues looked at physicians who were thinking about or talking with patients about prostate cancer genetic testing following a prostate cancer diagnosis. 92% did not feel comfortable discussing the benefits, risks, and limitations of genetic testing with patients. These are primary care physicians. 92%! Okay, y'all know, you're pretty numerous, I'm going to guess. That's, the most, similarly, 96% do not feel confident answering patients' questions. This is really frightening. 79% do not feel prepared to answer patients' questions, and 74% do not feel confident in their knowledge. Right, so we have a lot of work that we need to do with primary care providers so that they can be engaged with patients. And in fact, patients kind of understand that physicians don't understand this information. In a study by Sarah Golst, a former Ph.D. student at Michigan, she looked at people who had gotten direct-to-consumer genetic testings and asked if they shared those results with their physicians. The vast majority did not share their genetic testing results with their physicians, and the number one reason, they didn't think the physicians would understand it, or they wouldn't be able to interpret it, or be able to have a good conversation. So why bother? So, and as you might imagine, patients have an equally difficult time understanding this information. Who knows, and there isn't great evidence, at least not that I could find, about how well patients can understand this direct-to-consumer genetic testing or even their understanding of their genetic test results after speaking with a physician. And I know that there's been some work done in this, and again, over and over, you do find that there is this significant lack of understanding in the implications of receiving genetic testing results. So now what I would like to do is give you a couple examples of studies that I conducted with my team back at Michigan, looking at some of these factors. And one of the things that we were really interested in our study in the VA was people's understanding of the difference between a family history of a disease and a genetic cause of the disease. And in this case, we're looking at colorectal cancer screening. My favorite thing to do as a researcher is to look at risk communication. That sounded kind of geeky, didn't it? So a lot of my work, a better way to say it, a lot of my work really is involved in testing different ways of presenting information. So we thought maybe if we presented this risk information in different ways, it would have an impact on people's understanding and their use of the information. So we varied that, and as well as, sorry, I have a cold. Whether the patient in the scenario, so we gave them scenarios, had them read through scenarios, and then asked them what they would do if they were the patient in the scenario. And in that scenario, either half the people read about a scenario that had the mother had colorectal cancer, history of colorectal cancer, and the other half didn't have that family history. Sorry, I never drink water, but this is getting ridiculous. Okay, and then we randomized people to what kind of genetic test results they received. One is that they are told that the results indicated that they are average risk. So, and we clarified that even though you might have had a family history, in the case that it did, of colorectal cancer, it was not genetically caused. And then the other half of people were told that they are at high risk for colon cancer based on their genetic test results. And this is how we displayed it. Here's a nice pictograph or icon array, and here's kind of a thermometer. What we ended up finding out is that it didn't matter how we displayed the information. People understood it or didn't understand it equally well or equally badly. But what we did is then we did measure two outcomes: how likely you think you are to get cancer. But what I'm going to focus on today is our preference for getting a repeat colonoscopy, because the goal would be that if you're not at above average risk for colon cancer, and if you only have one family member with colon cancer, that you could still probably get a colonoscopy every 10 years. And so we wanted to see if people would be reassured by this information. And what we found is that patients with a family history perceived that they were more at risk than people without a family history, and that's good, because what that means is people are paying attention to the survey, and that you would think that if you had a family history, you'd feel more at risk. But we also found that people with a family history who received average genetic test results had a higher perceived likelihood of getting cancer than those with no family history and above average risk. So the family history here is really driving people's attitudes, and they weren't able to incorporate the information about genetic testing in their perceptions of their risk. In terms of their preference for receiving colonoscopy screening, what we found is that so again, the recommendation is that you get a colonoscopy every 50 years. Every 50 years. Wouldn't we all wish that? Every 10 years. But what we found is that even people who had no family history and had an average risk of receiving or getting colorectal cancer, so no genetic susceptibility, half of them wanted a colonoscopy more than every 10 years, greater than every 10 years, which personally I think is crazy, but but that really bumped up to 85% when people had family history. Now, and then you'll see the numbers here. When people had a family history, when they were told that they are at high risk, would be more acceptable, most people did want to get that screening more often. And just to kind of prove to you that there were no differences, this is what happened when we presented it with a thermometer. Now, what do you guys think? What do you think physicians had a different response? Do you think physicians understood this better or incorporated this information similarly or differently? Yeah, different. It's the same. So physicians also, about half of physicians still wanted a patient with a family history but average genetic testing to return about every 5 years. And so what I was trying to show with that data is that really people, family history, which is something that's much more tangible, something much more understanding, something that people are so much more comfortable with, probably played a bigger role in their decision making and their risk perception than the genetic testing results. Now that might be good or bad. I'm not going to judge. I'm not a clinician. I don't have that kind of expertise. But what I'm trying to really want to emphasize is that people are having a very difficult time understanding how to use that genetic testing result information, and that shows that we have a lot of work to improve that understanding. And the last thing I want to talk about is understanding the ethical implications of receiving a genetic test results. Going back to my little story, I actually had the genetic test done, and I didn't ask for the results for I think it was like a year, because I was studying this and I knew that it had a lot of emotional impact, and especially when it was unlikely that there would be one like really clear signal of what I should do. And so we wanted to look at how people understand information and how they feel about what kind of genetic test results people should receive. And so this work was done by my colleagues and my postdoc, actually the one who I texted, Michelle Gornick. And the goals here was to look at what, a lot of times we ask patients, would you want this genetic test results? And patients very, very frequently say, yes, I would definitely, I think this is really important, everybody should have this information. And what they wanted to do was to look at, when you educate people, when you really explain the risks and benefits of getting genetic test results back, will people's opinions or change about what kind of information should be provided to people? And so what we did here in this study, or I shouldn't say we because I was a very outside consultant, but what they did was do this process called deliberative democracy, which was they brought in about, I think it was 100 patients, or 100 participants, 50% had cancer diagnosis, 50% had not, and then they brought in two experts, a bioethicist and a geneticist, to really explain genetics and the ethical implications of it. Now, before any of that education happened, they did a pretest asking people about 3 different types of genetic test results. A medically actionable result, but was outside the goal. So let's say you're looking to see if you're at a higher risk for breast cancer, but you find out that you're really high risk for diabetes or genomics, and you could act upon that. Or let's say diabetes, and you could start taking metformin or whatever. So it's medically actionable, but it wasn't the goal of the test originally. Should you provide that information to people? The next was, what if you're doing like with my child and you find out that he has a high likelihood of getting prostate cancer or colon cancer 30 years down the road? Should we provide that information to the patient or the parents at that time? And then finally, carrier status. Are you a carrier of a disease? And so then so we did pre-test of that information, asking people how they felt about what information should be provided. And then after they did this big educational intervention, explained stuff, and had the patients talk together the participants talk together about the pros and cons of providing information or withholding information, we did a post-test and we looked to see how that changed with education. And what we found here is that in the medically actionable, there was absolutely no difference. People basically agreed with the default, which is that information should be given to patients. The other two, you find a huge difference in terms of giving, withholding information from parents about child onset things that would be adult onset. At first, only 10% of people agreed that you shouldn't tell patients that or tell parents that, and then that number went up to almost 50%. Same thing with carrier status. Only a small number of people thought you should ever withhold that information, especially if there's nothing you can do about it, and then that went up to 20%. And so what the implications of this is when people have appropriate knowledge, they may change their view on the appropriateness of getting information. And so a lot of times we do public surveys and say, what do people actually want? Well, if people don't actually know very much information about genetic testing or any other topic, it's really important. It might not be good data, obviously. And then also you'll see that there's a huge significant variability in people's opinion regarding what information should be provided and not. So there is, and that adds its own complexity. So there's so many questions we still need to answer in this area. How do we provide, what kind of risk communication strategies can we employ to improve patients' understanding of the information? How do we develop better genetic test results so when we're sending them to primary care physicians and we're providing them to patients that they can actually understand it? And how can we help physicians how can we train physicians to better communicate this information to their patients so that they don't say, well, I can go find the number for the genetic counselors if you want to visit them. Similarly, we have to understand how patients weigh and trust different pieces of risk information. I've shown in terms of breast cancer, when we give people Gale model scores and what their risk of breast cancer is based on the logarithms, about 20% of people say, I just don't believe your information. I think you're wrong. I'm special in one way or another. And they just don't believe it, and it could be because they don't trust us or they just don't trust science or a number of other different good reasons. And then we also need to figure out how to better engage people in decisions about what test results to receive, and not just because their friend is, or they heard it in the paper, or Angelina Jolie did, or something, or those kind of factors. Thank you very much.
All right, we've got about 10 or 15 minutes for questions. John?
Thank you. My question is for Kimberly. I was very interested in the conclusion of your talk. Would you favor actually giving consent orally with, say, a recording or a videotape as perhaps an improvement over a signed consent form?
That's a good question. I think of it more as a combination. So using a simplified form but not relying on it in the way that we sometimes do. I think for the educational component, relying more on verbally delivered information or multimedia information, and not relying so much on the form or written information to give the background information that people need in order to make an informed decision. So I think the answer is both, but using it somewhat differently than we are in a lot of standard procedures now.
There's comment slash question is for the first speaker, but it might apply to both of you. So you showed the use of pictographs, which shows stepwise what's being done with the samples for biobanking. I recently saw some demos at AMIA. It's a medical informatics conference where people created cell phone applications for regular people to use to see if their children had signs of autism spectrum disorder. And the consent, it had to go through an IRB also. So the way the consent worked is they would see one step at a time, and then you press next, and then the next step comes up. It's like one short sentence with a picture and so on. So they could keep clicking next, 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 and finally they give consent. But it also had an option for, the people in those diagonal box category. If they wanted to read the long version, they can press a link and read that. So it kind of reduced the information load to 1 byte at a time, so people could, consume it easily. So maybe I thought, technology could be something that helps here. And
just to respond, I think that's a great comment. This was at my previous institution, and I was not allowed to do anything other than a written form, so I tried to make the form as good as possible, knowing that there were no other options. But I think we can make better use of technology. Breaking information into manageable chunks is actually a fairly important thing, organizing it and giving it in manageable chunks, and I like that model a lot.
Another comment about the genetic testing results that you showed. It had so many rows of all these different mutations and steps, and it says unknown clinical significance, blah, blah, blah. It seems like it's a poor, for the lack of a better word, user interface design. People care so much about user interface on computers, but it seems like paper gets overlooked, because they could just as easily classify things into these are the results that are clinically significant, and then there's this whole other section. We don't know what to do with these, correlate clinically or something, and then list a whole bunch of things. But not only that, there is no point in really presenting all those mutations with unknown clinical significance unless either the lab, or if they have an intelligent longitudinal medical record, if it would periodically send a notification to the primary care provider saying, hey, we found this new information about the significance of this mutation that your patient was discovered with a year ago. So it seems like there needs to be longitudinal follow-up as well as some user-friendly presentation of data, rather than printing out a spreadsheet in rows and columns and throwing it at a clinician who hardly has much time to care for the patient. So anyway, those are the things I want to comment about.
And actually, and I think there's two different things about the unknown clinical significance. I mean, that's a question, right? Like, is there any reason to present that information? Is it nice for me to know, and so that I can keep my eye out in the newspaper and journal if it's P28, I think it was. If they find a finding, then I can know about that and I can search for it. Or is it just a worry? And I don't know the answer to that question, but I think that's something we need to explore. I think we have this knee-jerk reaction that we need to always present every piece of information that we have to people. More information is always better than less information. And I don't think that's true, and I think we need to be more thoughtful about what information is actually helpful and what information can be harmful and have absolutely no benefit. And in terms of the forms, you're absolutely right. And actually, my postdoc, Michelle Gornick, is redesigned it with some graphic designers where the important information is at the top. It says, what are the actionable results? And then all the gobbledygook that people who understand it can understand it is there, so that people who want to have it can have it. But that right in that first, just this little bit apart, it tells you what you need to know and what you need to do with this information. So it makes it very clear for the provider and for the patient. And she's about ready to go into trial with it to see if what the implications of it, if it improves knowledge, understanding, risk perceptions, worry, all those kind of factors. It's a great point.
And if I could just add one thing to that, I think one of the other things we've talked about here at Utah with Louisa Stark and others from the Genetic Science Learning Center is the need for educational initiatives that target both providers and patients, because I think we think a lot about patient-oriented education, but not as much about education of primary care providers and other users of genetic information. And so trying to get those two types of education going in parallel.
Well, and I think HIPAA 2 is breaking down that traditional distinction between information for the clinician and information to the patient, because increasingly patients are going to have direct access to laboratory information that needs to be probably written in a way that they can understand. Yes, sir.
They all like it, and some better than others. So you'd obviously want to use whatever was best.
So I think the question was, how do you know you have a good genetics lab?
You know, and I think that's the difference between, I think that is an excellent question, and I think that's the difference between when your physician orders that you don't really know that, you don't have any kind of control over where they send it versus, maybe to this, and not that I'm advocating for this, believe you me, but the direct-to-consumer advertising, they have data, you would like to think about how awesome they are, but you could potentially do a comparison there, but that's a little bit unlikely, I would say. But I don't think a lot of times you have control. It's where your physician sends it. It's where the hospital has the contract with.
Yeah.
I wanted to make one comment and then a question. So I really actually enjoyed the last question from this gentleman about ways we might actually drill down on the quality of genetic testing. And as you know, there's a tool developed here called IOBio that's actually trying to bring that technology to the bedside and to patients. And it seems to me that question actually spans the excellent presentations that I enjoyed hearing. I was wondering if you might speculate briefly on some other ways that we might drill down on tackling the issue of how to help gain the trust of patients who do get genetic testing in believing the data, and then also identifying areas of coverage that may be limited, where perhaps we may need to drill down in more detail there before we call a variant a variant.
I think those are both really good questions. I've done, and not easy to answer, but I think I and others have done some work looking at how to convey the idea of sort of limitations of genetic testing, so what genetic testing can tell you and what it can't tell you. And I think my major message from that is people are just kind of surprised to find out that there are limitations, that, it's not a definitive answer. But in the various populations I've worked with in Detroit and St. Louis and so forth, they've been pretty open to that idea once you raise the issue with them. So I think it's not, it might be more a familiarity issue that there are limitations, because we don't tend to talk as much about limitations of testing as much as its benefits. And I think raising trust is a difficult issue, and certainly I've worked with in my own work, but I think sort of the more long-term relationships that we've talked about in some of the prior sessions are really necessary, not going in one-off, but really trying to develop more relationship with communities. And I don't know if you wanted to add.
The only thing I would add to that, and I know, and it's what you were talking about in your talk, is that a lot of times we develop these tools or these consent forms without any testing it with the users. And so we don't know what people understand or don't understand, or what could be perceived as untrustworthy versus trustworthy. Things that we thought would be brilliant, when we showed it to the actual patients, they were offended by it. I mean, that doesn't happen often, happily, but a couple times it's like, oh, whoa, that was really taken exactly opposite. And I'm so glad I did this with, the ladies in the cafeteria who served us, rather than the first time we interacted with the patient. So I think really being thoughtful about those things before you start interacting with the patient is really helpful.
Leslie?
So one of the themes in the prior session was about communication over time and retaining samples over time. And I would assume that if people take consent forms home with them, they may forget about them, lose them, unless it was something highly salient to them. They might forget about the whole thing, and then data reuse might come into play. And I wondered if you've either done any work on or have any thoughts about how to achieve communication across time, which is going to become critical with the long-term cohorts.
The study that I talked just very briefly about, where we contacted women who were biobank participants who had signed up for a biobank within the last 3 years, I think the single most alarming thing about that study was how few of them remembered that they had signed up for a biobank, and that was actually a relatively highly educated population. It was just, this was one more thing that they did in the context of being diagnosed with breast cancer. They were overwhelmed at the time, and they just didn't remember. But in a lot of my work, people really want information, not necessarily even their individual information. They want to know what's going on. And I think the suggestions have been, newsletters, online sort of websites where people can go to. I think it's not necessarily that we need to spend a ton of time developing really individualized messages. What I've heard certainly across studies is that people just want to know what's going on. They want to feel like they contributed to something and that something's actually happening with their information. So as a result of the biobank study that Dana-Farber started sending out, I think it was a quarterly or semi-annual newsletter that talked about what was being done with those biospecimens, because we weren't allowed to return individual results. But even that really seemed to make people feel like they were a part of something and remember, frankly, that they were in a biobank in the first place.
Yeah, that's exactly what I was going to say. And then one thing I wanted to add a little bit about that is we had colleagues who was looking at informed consent and people's reading of it. And, there's always the understanding component, but they put in like one sentence in there that says, if you actually read this sentence, click here. And nobody clicked it. And it was right in a paragraph where there was really important information. So that's also a huge concern about this, is when we do these consent forms that are long and complicated and boring, people don't read them, and that in itself is a huge issue I think we need to be aware of.
Well, in terms of time, too, one of the things our group is working on is trying to get the consent and education process out of that compressed interaction between the clinician or investigator. You can send them the consent form on their iPhone a day or two beforehand and say, if you have an opportunity to read this, and then we perhaps can talk about this when you come into the clinic. You have presumably a more informed dialogue at that point. Getting it out of the clinic, which is a terrible place to have these types of conversations. I think we have time for one more question. Yes, ma'am.
Have you done any studies that analyze the ethical implications of getting genetic test results on more than the patient or the parent of the patient, but extended family and things like that? If one sibling gets the testing, how that affects multiple people and generations and so forth?
I actually have not. Have you? Okay, so the thing, I just want to make sure. So what I've read a little bit about this, and I will admit this is not a topic that I'm well read about. But there's a lot of implications to this, right? So some people, especially if you think of siblings or cousins, that some people don't share it, and some people don't get tested, some people do. Some people get tested and don't share it. Some people test it and do share it, and that can actually lead to a lot of familial discord, which is a concern in itself. That very rarely, when you get this genetic test, is it only limited to one person, right? When you think about breast cancer, BRCA, those kind of, Huntington's disease, those kind of especially really prevalent ones, they're going to influence a lot of other people's decision makings and emotions and fear. And I think that's something that also needs to be really well communicated to patients, that, without scaring them to say, oh, well, I don't want to do this because what if it affects somebody else? So how do you walk that tightrope so that they're aware of that as a factor of their decision and outcome of that decision, but without them having to worry about making a decision for everybody else?
Please join me in thanking our speakers for their presentation.
Why don't we do a quick changeover to our final session for today. Also, while we're switching over, I want to assure our law students that are, volunteering in the audience that when you hear people talk about documents that are overly legalistic, that we don't consider that a bad thing here at the law school. So it's all a matter of perspective. Anyway, Leslie? That's right. Legalistic is, you get an A if it's legalistic. Oh, I guess not a USB slot, so we can get it in there.
Meyer's got hers in one too.
Oh, really? Oh, okay. So actually, this is good. So which is the presentation?
Robert's Utah Precision Medicine final.
Final there.
That one? Okay.
We're going to make that executive decision.
So Roberts, okay, that's also on panel 8.
Yeah.
I'm going to just put
I was
8 there.
guessing we're sitting. Let me ask Maya.
Because now it's in the box. Okay.
I'm gonna.
And then we have somebody else?
I'm not gonna need the whole time. So,
Okay.
yeah.
So
I will be good. I will
degrees in political
not
science,
even be a
right?
challenge for
You
you.
have degrees in law and political science. Yeah.
It's 15 minutes, yeah. But I'm probably only gonna be like, I'm not.
I didn't
title it stuff until you want me to title it for me.
I can do that, right? I can do that.
Okay, we can open. I'll open it in a second, and then we can.
Okay. Oh, that was the wrong one. Okay, here we go. Okay, here we go. Is that one Utah presentation? Okay, I'm going to put an 8. No, we want it in the box. No.
We'll be getting going in 2 seconds.
Okay, are you first up, Leslie?
Yeah, I'm going to just talk for 2 seconds.
Okay, no, you're, okay, we're good.
I think we're going to stand.
Are we going to stand? Yeah, why don't you guys sit over there for just a second and
Can I
I'll get it
grab
going.
Mike and
Sure.
Mike?
Okay. Yeah, it's all up.
So we're gonna. Okay.
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 So thank you all for staying with us. I'm Leslie Francis, and I'm going to be moderating this panel on precision medicine, the ethics of access. I wanted to just take a couple of minutes to outline some of the challenges of access, which has been an ongoing theme all day today. It's not only whether people are going to have the money to pay for expensive therapies, it's also whether any of the promise of more accurately tailored treatments are likely to be available to groups when there's limited information about people who might fall into those groups. Just to do a little more of the dismal data about access, this is from the report of the NIH working group on the Precision Medicine Initiative and putting together the Million Lives. The goal is to design the cohort to have sufficient statistical power to detect differences among major demographic groups in the U.S. These groups to include not only race, as in the census categories, but also sex, age, geographic, and socioeconomic differences. Special policy attention is to be devoted to inclusion of people who can't give their own consent, such as children and people with intellectual disabilities, prisoners, or people who, while they start out capable of informed consent, may either lose decision-making capacity or find themselves in situations where it might otherwise be considered inappropriate to enter them into clinical studies like prisons. Just to give you a little bit of a feel for the persistence of huge disparities, I think that's all we can call them, in participation in clinical trials by race and ethnicity that have persisted over time. 2004, one of the most cited studies in this area by Murthy, Krumholtz, and Gross, they found significantly lower numbers of Black and Hispanic participants in National Cancer Institute-sponsored trials, with rates very disturbingly diminishing between 2000 and 2002 2002 was the year that the Institute of Medicine report on race and healthcare came out. So this is in the 2 years preceding that. In the 2015 PMI initiative report, African Americans make up 5% of clinical trial participants, and Hispanics only 1%. And I've got in parentheses their population numbers. So I just had a little fun. I went to the FDA website. They have a nifty new tool that's designed to tell you whether newly approved drugs might be good for you. Okay, that's kind of how it's billed. It's called Drug Trial Snapshots. It gives demographic information by census group categories about patients included in clinical trials of newly approved drugs, and considers whether the drugs, whether the trials contained significant, contained enough information about groups such as African Americans or Asian Americans or Pacific Islander, Native Hawaiian, or Native American, Alaskan Native, to draw any kinds of significant conclusions about either benefits or risks. There were 78 new drug approvals in the last 2 years, 2 of them with data that enabled any conclusions to be drawn about African Americans, 7 with enough data to draw conclusions about Asians, 2 more with data that allowed some conclusions about both blacks and Asians, and 9 with enough information for more across-the-board significance. But 58 of the 78 approvals, when you go to the website and you want to know whether there's anything known about whether there might be risk or benefit differences in your demographic group, you are told we don't have enough information. Amusingly, Tresiba, which is the Novo Nordisk new blockbuster, super advertised diabetes drug, was one of the 58. So I'll just let you think about that. And without any more ado, I want to introduce our 2 speakers who are going to talk about this much more than I can. First, Jessica Roberts, who's director of the Health Law and Policy Institute in the law school at the University of Houston, where she's also a George Butler research professor. And Maya Sabatello, who has degrees both in political science and in law, and is an assistant professor of bioethics at Columbia University. She has a long record of working on issues in human rights and is co-chair of the American Society for International Law, International Disability Rights Group. And without more, I'm looking forward very much to hearing from Jessica and Maya.
You have the clicker up here?
Do we have the clicker? Yeah, it's on the...
Somebody stole the clicker.
That's okay. Okay, so thank you so much. Thanks to the organizers, especially to Leslie Francis, for the opportunity to be here today. And I'm excited to talk to you all about what I call upstream access issues in precision medicine. And in fact, we've already heard a lot about these issues today, but I don't know that we've necessarily thought about them in terms of access. So, to give you a sense of what I'll talk about, I will briefly go over the goals of precision medicine and the various kinds of access issues they present, and then I'll focus on these upstream access issues. And what I mean by upstream access issues is I mean access issues that deal with research design and not with end users. And so these kinds of upstream access issues raise at least two questions for us to consider. First, what should we study? And secondly, how should we study it? And then I'll end my presentation today with some takeaways regarding how to ensure all Americans can have equal opportunity to benefit from precision medicine. So, as we all know, remedying health disparities has been a major focus of healthcare reform in the United States, and people experience health disparities based on a variety of different factors, so race and ethnicity, national origin, sex and gender, disability, age, geographic location, as well as socioeconomic status. And we must make sure that in the move towards precision medicine, we do not create or exacerbate any of these health disparities. And so here I have for you guys on this slide a quote from the National Institute of Minority Health and Health Disparities, which is part of NIH, doing its own precision medicine research. And they tell us that while precision medicine holds great promise for improving patient care, its potential for reducing health disparities hinges on better understanding of the dynamic interplay between biological, behavioral, social, and environmental health risk and protective factors experienced across the life course, coupled with greater inclusion of health disparity populations in research aimed at developing precision medicine interventions. So we need to be concerned about not just studying disease, but in just studying the context of disease, as well as making sure that we're having representative individuals participating in health research. So in terms of the success of precision medicine, there are a lot of different factors, but what I am focusing on is I'm looking at issues related to funding. So which kinds of projects are we funding, as well as patient participation, which we've already heard a significant amount about today. So most often, as Leslie alluded to, when we talk about access issues, at least with respect to medical technologies, we're focusing on what I call downstream access issues. Right? So these are the kinds of questions we ask ourselves: how much is it going to cost? So are certain populations going to be priced out of precision medicine treatments and therapies? Right? Another thing that people consider, right, will your health insurance cover whatever the treatment or the therapy is? In particular, will health insurance policies under Medicaid and Medicare cover you? Because those types of insurance cover vulnerable populations or underserved populations, right? So Medicaid is the #1 provider of health insurance for people with disabilities, so whether or not you have coverage under Medicaid is something that's significant in terms of those populations having access. Also, you might want to consider where, in terms of geographic location, people manage to access precision medicine. So is this something where you can just walk into your family clinic and you can get access to a treatment or therapy? Or is this something where you have to go to some sort of fancy clinic that's somewhere further away that might be more expensive? And of course, in addition to these other concerns, there might be other sorts of significant non-monetary types of investment in terms of technology or time, right? So is this something that's going to require a large time investment that I'm going to need to take time off work? Is this something that's going to require me to have technology or access to technology like a mobile phone or the internet? And so these are all the kinds of concerns that folks, I think, frequently think about when we're saying, okay, what does it mean to have access to precision medicine? And as I mentioned, they speak to who can access the technology after it has been developed. So this is about end users. However, I propose to you all that the nature of precision medicine also opens the door for what I call upstream access issues, right? So as we know, precision medicine is something that is algorithm-based, right? We use all this big data that we've been talking about today to formulate rules and algorithms that then refer to certain reference populations. And the kind of data we collect and from whom can have an effect on how these various algorithms operate. And my point to you all is that not everyone may have an equal opportunity to benefit. So upstream access issues relate to accidentally building access barriers into the research and technology design unwittingly. So not all populations will benefit equally from the same research. Why? Well, because as we know, disease varies across population, right? And not all populations participate equally in research. However, if precision medicine only benefits certain groups or benefits certain groups disproportionately, that could be something that then creates or increases health disparities. So, to sum up, conversations about access to precision medicine have focused, at least in some regard, on these downstream issues, but we may unwittingly be creating access issues upstream by building access barriers into the rules, algorithms, and reference databases that actually make up the foundation of precision medicine. And to avoid creating upstream access issues, we must consider what we study and how we study it. Right, so in terms of what we should study, right, I had this quote here from Barbara Rossman, and she tells us that, quote, scientists are not detached observers of nature. They are constantly making decisions about what they will consider significant, and these choices are not merely individual or idiosyncratic, but reflect the society in which the scientists live and work. And we obviously live and work in a society that values precision medicine. Now, it's useful to take a step back and consider why that might be, right? Because as we know, disease is only one factor related to health. So I'm sure many of you are familiar with this graphic. This represents the social determinants of health. So we've learned that environmental and socioeconomic factors also have important health impacts. So when we devote resources to science with the goal of improving health, we're then diverting resources away from other potential uses that could also improve health. And by that I mean by providing housing or improving food security or even education. So precision medicine has huge potential for improving health. That's why we're invested in it. But the success of precision medicine depends on this level of sustained funding. And my proposal to you is that because we are prioritizing precision medicine as a society, we must ensure that we approach precision medicine in a way that ensures an equal opportunity to benefit for all Americans. So there's a lot of things that we could study in the context of precision medicine. Now, here I have the graphic of leading causes of death. Right? And then in looking at these numbers, it's no surprise that one of the initial targets of the Precision Medicine Initiative has been cancer. And if we look at how cancer is distributed across populations, this has already come up a bit today. It's not distributed equally, but we can see that several different demographic groups have the opportunity to benefit if we manage to eradicate cancer, that it will in fact hold some degree of benefit across races. However, that is not always the case. So certain conditions might be more highly concentrated in one population or another. So this slide has cystic fibrosis, but I could have picked any number of different conditions to demonstrate that certain diseases are more concentrated within certain populations. Now, I don't mean to say that we should ignore population-specific kinds of conditions, but we want to be aware that if that's what we're studying, we then go and study a condition that has an impact in another population. So if we're going to study cystic fibrosis, we then might in turn want to study sickle cell. And of course, something for us to consider is that precision medicine is not just about disease. So this quote here is from the website of the research cohort, the All of Us research program, and it explains that the research program is not just about disease, that it plans to focus on ways to increase an individual's chances of remaining healthy throughout their life. Well, of course, this brings us full circle to the social determinants of health and the concerns of the National Institute of Minority Health and Health Disparities regarding precision medicine's ability to account for the interplay of disease with behavioral, social, and other kinds of factors. So, to avoid creating and perpetuating health disparities, precision medicine must consider context. Also, too, while my slides have focused primarily on variations across race, I started by talking about all the different types of health disparities in the United States. So we also want to consider variation with respect to sex and gender, as well as age, disability, and income level. Also, so when we're talking about what we study, the things to keep in mind, right, is that precision medicine itself is a research allocation decision. And what we study with respect to PMI could have a disparate impact because not all populations will benefit equally, and that we want to achieve the goals, make sure that a majority of Americans can, in fact, benefit. So also, who participates in research? could also affect the opportunity to benefit. We've already heard today about the importance of a diverse research cohort to ensure the accuracy of research across populations. And we already know that certain populations do not participate with equal rates in the context of research, right? And we know that one of the reasons that certain groups might be less willing to participate in research is distrust. So this one particular survey shows that distrust is of particular concern to the Black community. Now, this is not hugely surprising, right? Given highly publicized accounts of unconsented research. So I'm sure a lot of the images on this slide are familiar to many of you. Right, we have The Immortal Life of Henrietta Lacks, which not only was a best-selling book, but now is being made into a movie with Oprah Winfrey in it. And so while it might be unclear whether or not the scientists actually violated the informed consent standards of the time, there was still widespread perception that this was some form of exploitation. Also, too, I have the Protect Baby DNA to represent the scandals over newborn blood spots, and I'm sure many of you too, over 5 million blood spots being destroyed in my home state of Texas. So that was something that was raised a number of concerns. Also, the photograph above that is Carletta Tolosi, and she was the lead plaintiff in one of the cases in the Havasupai litigation. So this was when a tribe consented to blood draws for diabetes. The researchers ended up doing other studies, including on topics that were taboo to the tribe, including mental illness, inbreeding, and migration. And then, of course, any conversation about distrust of research is happening in the shadow of the Tuskegee syphilis study, right? So a lack of diverse research cohorts could compromise the accuracy of precision medicine for certain populations, which would be, again, another one of these upstream kinds of access barriers that I'm describing. So what should you take away? Well, a couple of things that I'd like you to get from this presentation. One is that we need to be thinking about these various concerns we've heard about all day as upstream barriers to access, and when we need to think about them in conversations about access, in addition to the downstream barriers people might encounter, and that even providing meaningful downstream access might not be enough to avoid creating or perpetuating health disparities if we've built in these discriminatory mechanisms upstream. Right? So going forward, we need to consider both affected populations and the relative accuracy and effectiveness of precision medicine treatments and therapies. And so for the future of precision medicine, I think that we need to think deeply about the kinds of research that we're funding in terms of making sure that we're providing robust funding for conditions that vary across a number of different populations, and that we need to make sure we are building trust in science so that we're able to recruit diverse research populations. And that is it for me. Thank you very much, and I will pass it off to Maya. Maybe not. Can you show everything? The presenter one? The presenter view? Is this your one? Yeah, that's mine. Oh, yeah, okay. But the presenter view within that. Do that. Yeah. There you go. Wonderful. Thanks. That I didn't do. I tried. Hi, everyone. Thank you so much for being here. I know I am the one who is holding you off from actually having the reception, but I do want to say, first of all, thank you to Leslie and to everyone who organized this symposium, which has been really wonderful so far. And I'm really happy to be here as well. So thank you very much for the invitation. I'll be discussing precision medicine, disability access, and ethics, and I think it really builds nicely on the presentations that we've heard today. It also really comes into a lot of the work I've done before. I've done a lot of work with regards to disability rights, and I'm also co-directing a project at Columbia University on precision medicine, ethics, politics, and culture that brings in a lot of sort of interdisciplinary discourses, including in the context of precision medicine and disability. So, of course, precision medicine programs are gaining momentum
across the healthcare landscape in the United States. A recent study found that about 30% of 137 facilities that have been studied in the country are conducting at this point precision medicine programs, and which is of course a significant number, and it has increased in the past few years. Obama's announcements in 2005 of the Precision Medicine Initiative, or the All of Us program as we now call it, has been instrumental in this regard. First of all, in making it a national priority to have precision medicine programs, but also in terms of the commitment to the budgetary allocation for such programs. So that's been quite significant. And in fact, since the announcement of President Obama, what happened was that there's been an increasing discussion about the need for racial and ethnic diversity in cohorts, simply because if we want to identify genetic variants that affect disease risk and drug response within and across groups, we need to have that great diversity within the cohort. The other sort of promise of precision medicine, definitely on the national level, is the idea that it will help to curtail some of the health disparities that exist in this country. Again, it will become available to larger groups that at this point have significantly worse health conditions. But the place and role of persons with disabilities in precision medicine programs has not received much attention, and I think it does require the question, first of all, why? Considering that we're talking about quite a large size population in the United States, 22% of adults are considered or classified as people with disabilities, and 13% of children. The other question which I would like to kind of discuss today is whether and why including those with motor, mental, cognitive, sensory, and intellectual disabilities should be a part of the precision medicine agenda, again, both in smaller projects but as well as on the larger national program. So what I want to do is look, first of all, into the relationship between disability and genetic research, highlighting the possibilities and concerns that this intersection arises, and I think that they will inform our considerations and discussions also as we move forward with precision medicine. I will then look into, make the case for disability inclusion in precision medicine projects, focusing in particular on scientific and social justice rationales, and then moving forward, thinking about how to actually ensure disability inclusion, taking into account questions of access and ethics. So, disability and genetics, of course, there's the logic that connects genetic and disability seems to be quite straightforward. We think about genetics as a field of research that aims to explain how molecular maps are expressed, that is, how genotypes impact phenotypes, and including, of course, how what we view deviant phenotypes are impacted by genes. So that brings us really straightforward to disability, which has been, again, a major area for genetic research. But beyond that, of course, genetic research can be for the mere endeavor or interest in increasing general scientific knowledge, but it has been mobilized, this entire movement of genetic research and disability has been mobilized by the therapeutic imperative, that is the drive to prevent or cure disabling impairments, with a range of applications that are currently used or under development, including prenatal genetic testing, pre-implantation genetic diagnosis, postnatal predictive and diagnostic genetic testing, which we've heard quite a lot about today, pharmacogenomics, obviously in the context of cancer, that will be very important, and gene therapy that is currently in experimental stages, but again, we might be thinking about it for the future. But the application or potential application of genetic research and knowledge has raised three main criticisms from the disability community, which again I think are likely to inform our future discussions also with precision medicine. So the first one is the concern that prenatal and pre-implementation genetic diagnoses are discriminatory or are resembling eugenics. There is a concern that it violates the right of future people if they lead to disability-free genetic selections. that it unjustly distinguishes among individuals on the basis of their genetic makeup. And there's also a slippery slope argument here. That is the concern that focusing too much on the genetics basically, and allowing, of course, such disability-based selections will lead to devaluation of persons with disabilities, to low tolerance for human diversity, and to the eradication of difference. And of course, the Nazi regime, the eugenic ideology and program under the Third Reich was kind of a very strong illustration of what can happen when we're trying to eradicate bad genes. That's actually a picture that I took just last week from Munich. I was in the documentation center there, and of course that's the Nazis' propaganda against people with disabilities. The second line of critique is that geneticization of impairments, that is the effort to find genes for all the type of impairments, divert attention from, and of course budget allocation from other social, economic, and environmental factors that cause or contribute to disability. And of course one of the findings is that today most of the disabilities are in fact not genetic based, at least not to our knowledge. They are a result of car accidents, wars, and so forth. And then the third line of critique is that disability, that the concern on, again, this ongoing search for genes tends to view disability as something that is very clear-cut, what's disability and what's not, as well as disability as a group is a homogeneous group, whereas in fact neither of them is true. And again, one of the most common examples is the deaf community, where they view their deafness as something to cherish, to be proud of, rather than something to eliminate. But might precision medicine be different? And I think that looking at the components of precision medicine suggests that the answer may be yes. Again, precision medicine looks at more than just genetics. It looks into environmental and lifestyle choices, which would include also socioeconomic status and so forth, which suggests that if it is implemented correctly, could be much more attuned to a social model of disability that puts more emphasis on the societal and other barriers rather than the physical or the genetic perspectives of disability. A second thing is that precision medicine, unlike previous disability genetic studies, which really focus on how to eradicate disability, precision medicine actually may offer therapeutic benefits beyond the impairment. So quite a bunch of the concerns that might have been raised in the previous studies of this kind of trying to find only the genes for might be somehow less strong. And so what I hope to show in the next few minutes is that the inclusion of persons with disabilities in cohorts fit squarely with both the scientific and the medical social justice goals of the precision medicine, including in particular the precision medicine on the national level. So scientific rationales. What are the scientific rationales? I think that persons with disabilities tap into all scientific measures that impact risk disease and drug response. Of course, it's a universal phenomenon, cutting across age, sex, race, and ethnicity groups. And usually what happens is that if race, ethnicity is one level of kind of deprivation, disability is a second one. If you are a woman with disability, you add a third level of deprivation. So it's something that kind of builds in to creating disparities. Another thing is that people with disabilities have high rates of comorbidities, almost by definition, or sometimes by definition, and they're particularly relevant in the context on some of the interests of precision medicine. For example, we have some studies suggesting that the risk of epilepsy and Parkinson among persons with intellectual disability is significantly higher. With epilepsy, they're talking about almost 20% versus 0.8% in controls. Parkinson is just about twice as much. Wow, just 5 minutes, which I was told 20 minutes.
You can go 5 more. Yeah. Sorry.
So that's, of course, important for us to know, given that 38% of the precision medicine projects today focus on neurological conditions. Same thing, we know that there are some studies suggesting that people with schizophrenia have increased risk for colon and breast cancers, but lower risks for other types of cancers, for example, melanoma, which again comes very nicely with the 80% of precision medicine projects today that focus on cancer. But I think that there are also social justice rationales that should be considered, and that is that persons with disabilities cannot be equated with patients, but at the same time that they experience unique barriers in healthcare. We have a study of over almost 2,500 primary health care facilities in California that found that only 34% are fully accessible, and that the number of facilities that are actually accessible in terms of equipment is significantly lower. It's less than 10%. We also know that persons with disabilities are less likely to receive cancer screening, which, as we heard today, that's a primary way sometimes for precision prevention. And that they are significantly less likely to participate in clinical trials. That's one, because they have higher comorbidities, which is usually used as an exclusionary criteria, but also because IRBs are often very conservative and are concerned of including people with psychiatric or intellectual disabilities in such studies. And then finally, we have a U.S. national study finding that people with disabilities are less likely to have private insurance and more than twice likely not to afford treatment also when they need it. Again, that has been discussed today, can have significant impact on enrollment in precision medicine efforts as well as in clinical trials. But overall, I think that's an important point to remember, that is disability is an unrecognized health disparity group. We're talking about health disparities in the context of race and ethnicity as something that today is clear and obvious. But people with disabilities as a group fare far worse than their non-disabled peers across a broad range of social determinants of health. And again, if you look into some of the statistics, which I'm not going to get into, they have very high rates of poverty, more than twice likely to live under the poverty rate, low employment and education statuses, and lower use of internet, which are quite significant, especially if you think about the increasing use of internet and all the gadgets that we have for precision medicine projects, for participant engagement, for learning about your genetic results, and so forth. So these are things that will be quite important, leading me to suggest that the inclusion of persons with disabilities is critically needed for precision medicine to fulfill its promise of curtailing health disparities in the United States. So some thoughts about moving forward. Again, given that precision medicine gives a lot of hope and it is a very promising program, I think we have to think about how should we go about to ensure disability inclusion and how can we do it in an ethical way. So I want to suggest just a few ways, obviously not comprehensive, but just some thoughts. One is creating accessible information. Again, we had here some great conversations before, but Braille, plain language, all of that, that's essential. We will never be able to get informed consent properly if we do not address those issues. People who are blind cannot today know what precision medicine initiative, including that is the All of Us program, actually means without that type of information. We have to think about the needs for involvement. Again, there's been some discussion about how to ensure the involvement of people with psychiatric issues. I think that's a big issue. What I've noticed being on the IRB, genomic IRB, is that there is a tendency of assuming that with people with psychiatric disorders or intellectual disabilities, we should just be enough with surrogate consent. The actual process of assent of people with those conditions is sort of minimal because researchers do not have the time, they do not have the funding, they do not have the skills, they do not have the expertise. But again, I think if we want to have these participants engaged and in the long run they have to be involved, we've got to figure out how to do that. A third thing is how to re-up benefits, which clearly requires address. I think first of all is, of course, access in terms of enrolling in precision medicine projects, which has been discussed, and I think I've made it clear here why it's important. But there's beyond it. Current precision medicine projects mostly offer return of results. That's including the All of Us program. But that's not enough. Again, we've heard that returning results does not clarify how to deal with them, with these results, does not provide information or interpretation about what these results mean, how you can actually act upon these results, which in the United States has a significant context because of the tendency of assuming that health is a personal responsibility. And I would suggest that we're increasing it here. We're moving into a genomic responsibility where we're providing genetic information to participants, hardly telling them what they've got to do, but once they'll get sick, there'll be a tendency to blame them, and of course insurance companies will be thrilled to add a premium for that. So I think that's another thing definitely to rethink, and how are we going to deal with that. And then finally, question of availability, accessibility, and affordability at risk. I'm just going to remind everyone about the EpiPen scenario and saga. Precision medicine therapies will be expensive unless we're going to make sure that they're regulated, they're affordable, that healthcare insurance are required to provide them. Otherwise, people who will be part of this precision medicine project, including in particular people with disabilities who are among the most impoverished populations in this country, will never have access to those benefits. So finally, I just want to conclude that policy guidelines should require engagement throughout the process, and it has to be with an open mind. That is the idea that we want to think about the possibility that some of the suggestions of people with disabilities are in fact correct, but also that we have to be open enough to hear what are their needs, what are we able to actually do to ensure that they participate, and they are indeed a part of all of us. So just a quick conclusion, precision medicine research can offer significant health benefits for the U.S. population. I really do believe that. I do think that achieving this goal and upholding the promise of curtailing health disparities will be impossible if persons with disabilities are not included. But how to do so requires preparation and ongoing engagement to ensure that efforts include beyond genomics. Again, we don't want to just go to the genetics and disability, which will raise criticism. We want to focus on all the social, economic, and environmental factors, and that to ensure that access to precision medicine research and therapeutic benefits is available to all. So just thank you very much, and we're obviously happy to open for questions.
We have a good 15 minutes for questions. Over there.
Thank you for the presentation. It was very enlightening. My name is Chedaj Kumar Singh. I am from India. So given the diversity of population and many other issues, my question is to both of you, I think. So it is about the accessibility. Given the nature of collaborative and open, biomedical research, when we talk about the accessibility, initially, as we have seen in the Human Genome Project, it was successful, as mentioned earlier, that because it has not attached to a viable business model. But when we talk about the precision medicine, it has a commercial aspect, and that is very much in conflict with the accessibility. So, have you, because you are working in that very area, I'm just asking whether the commercialization of precision medicine has produced some unethical, accessibility questions, and how you consider that. Thank you.
So, is this on? Or okay. So I think, so the question is, I think that in terms of the commercial nature of precision medicine and how that impacts access issues, and I do think that speaking to the downstream access issues in terms of pricing, right? So if you have something where a pharmaceutical company, has an exclusive right to a particular medication and can then price people out, right? We also have concerns in the context of health insurance, with price tiering for drugs and things like that. And I think that is a very real concern in terms of the commercialization of not just precision medicine, but just healthcare generally. Healthcare itself has been commercialized in a lot of in the United States, and so it's something I think that is a systemic problem. But it's interesting because there's actually the flip side to commercialization that I've seen. So some of my work that I do has to do with the ownership interest in genetic data, and so we've actually seen a move where certain genetic and genomic testing companies are interested in offering individuals commercial rights in their data for sharing, and so they're saying, hey, buy this genomic, there's one company that offers to test your genome for only a few $100s. They don't actually analyze the genomic information. They give it to you, and then they give you the opportunity to broker agreements with researchers. So you can say, hey, you want access to a, 36-year-old white woman's genomic information? Well, I can send it off, right? And so it tries to pair people and give individuals the opportunity to commercialize. And so I don't think in the United States we'll see a complete move away from the commercialization of healthcare anytime soon. But what we might see is allowing individuals the opportunity to themselves benefit commercially from participation in research, and that's actually something that's happening somewhat with some of these private genetic and genomic testing companies, and that might then become a more popular practice.
I just want to add that I think, there are ways of giving access without giving up commercialization. That is, you can still have some drug or pharmacogenomics or anything that will be helpful and give priority to those who participated in the research. For example, that doesn't cost you too much. It just makes sure that people who participated and they contributed to the public good, get something in return. I think this is kind of very simple way of doing it. So that's one other option.
So part of what I found both fascinating and important about your talks is that they both sound a cautionary note to how, if we don't design it right, precision medicine risks increasing disparities rather than, or in some ways entrenching them rather than improving them. Right. And the primary form of attention that's taken has been to try to be inclusive of what I think I would call census categories in the precision medicine initiative. So let's make sure we get a sufficient number of African Americans. Let's make sure we get a sufficient number of Asian Americans, etc. With some recognition that although the kind of census categories that are in the drug trial data don't include Hispanics, maybe for various lifestyle reasons or dietary reasons, that kind of ethnic subcategories matter as well to the PMI. But what both of you are talking about is a lot more than that. It's questions like making sure that we have access to getting into the system whereby data can be collected about you, the worry about exclusion criteria, and for Maya and for Jessica, the concern that if we study, say, diseases that have high prevalence in certain subgroups but not in others, we won't get an equal distribution of the promise. And I wonder if either one of you, or I would actually invite members of the audience to comment on this too, because I think this is the heart of what's a hugely vexing problem. Whether you have any thoughts about, you've got recommendations, but whether from a public policy pressing perspective, how are we going to get these into the NIH? Is a sort of funny way of putting it.
So I think, first of all, that there are two different types of accesses, right? The first access is inclusion. The first access is enrollment in different precision medicine studies, and I think NIH has done a great effort to include now ethnic and racial minorities, sexual minorities, Native Americans, and so forth, and I think they're really trying to do it hard. And I don't think that all the precision medicine projects in the country will be able to do the exact same thing. I'm not sure that all of them have to do it. But I think we have to think about what is the Precision Medicine Initiative or the All of Us program, for example, is really aiming to do. Is it only for scientific purposes, or is it to curtail health disparities? If the latter one, we have to put much more effort about recruiting this diversity that we need across the country. The other type of access, which I think can be addressed through regulations and some policies, has to do with how to actually enjoy those benefits. And I think at this point, if we're not, there's practically nothing in the All of Us program except for return of results that guarantees any benefit. So that leaves the entire system, healthcare system of private insurance, high premiums, individual decisions of healthcare insurance, whether they're going to cover, what they're going to cover, who they're going to cover, to what extent they're going to accept the validity of those trials that we're talking about, that even if you got into them, insurance companies don't necessarily have to think that they're helpful enough to cover them. So I think these are issues that we have to think about, and I think we have to think about them early on. That is, the more we're going to wait and run with the precision medicine without those considerations, it's going to be much, much harder to incorporate them and include those groups later on. It's just the way life goes. We start running, we forget to stop. So I think before we run, we kind of have to rethink and really make sure that we have those two types of accesses in place.
And I think that, so picking back off of that a little bit, I think that given the newness of precision medicine, we have the opportunity to get on the ground floor in terms of how we build these various structures. So I see this conversation as part of a larger conversation we're having in the United States about forms of structural discrimination, right? And we're just now seeing how various policies and distributions early in the process in terms of, access to education or housing then can have effects further down the line, right? And so this is a situation where we can look at the structures we're building and make sure that we're being inclusive and we're being thoughtful. So in terms of my policy recommendations, it's just for us to have this out in the open and to just ask these questions. Okay, who are the populations that stand to benefit from this particular line of research, both in terms of the impact of the particular condition or the disease, and then also as well as Maya is saying, in terms of the technologies and the healthcare, right, with respect to health insurance and other forms of access. And I think this is a situation where there's a highly optimistic note, right? Well, we can do this as long as we're thoughtful. And so I feel optimistic about it, and it's just exactly these kinds of events and these kinds of conversations that are going to steer us in the right direction.
I actually want to add just to that last one. The All of Us program is really unique around the world. It's the largest and most ambitious program with precision medicine research that took place in the world. All the other projects are significantly smaller number of recruitments, smaller initiatives. It's all on a smaller scale. The United States can take the lead, and it will take the lead. So why not do it right? And I think we can. So I'm going to be joining your optimism with the hope that these things will be indeed addressed beforehand.
So lots of hands have suddenly gone up. Yours was first, and then Jeff's, and then I saw two over here.
So thanks for this. I'll try to be quick. I know there are a lot of questions. So let me frame my point this way. I think there's a fundamental baseline problem in this discussion we just had that you just raised, Leslie. And that is, you can think about personalized medicine as individualized medicine, in which case then you need to think about the individual as a member of the group, as you say, census category or, whatever it is. Remember, we've had these categories before, Jim Crow, based upon blood quanta and so forth. So we obviously don't want to move to something like genetic quanta, whatever that equivalent might be. And so the other situation is, to what extent do these census categories help us, or are we creating new categories based upon genetic typologies? And if we are creating new categories based on genetic typologies, then I think the question is, why are we doing that? Are we doing that to remedy past discrimination? Are we doing that to be more equal? And in order to be more equal, it's sort of like affirmative action. We need to kind of recognize certain backgrounds and therefore distribute the benefits in a way that's more representative. So I would focus on that typology. I think that's sort of a fundamental problem in precision medicine, exactly what these typologies are, what the categories are.
I think that's a really, really great question. I think there might be a need to rethink about the census categories, simply because they don't necessarily reflect genetic ancestry, and we know that there are some problems with that part. kind of bringing together identifying race, ethnicity versus the genetic ancestry of some people, because self-identity is more social and politically construct versus the genetic one. Whether we should take it as a sort of to remedy past or current problems or discrimination, I think that's a nice idea. I actually love that personally, but I don't know that that will be endorsed, because we know that structural discrimination has been very hard and very difficult conversation. But at least moving forward, we just have to think about it in terms of ensuring equality for everyone from now on.
And so I understand your comment in terms of what people in anti-discrimination would refer to as the dangers of essentializing, right? So we're going to take these categories, we're going to imbue them with meaning, and then we're going to create meaning for the categories in a way that could then have some kind of adverse effects further down the line. And so I think that is some kind of a real concern, right? We don't want to replace other types of disparities with some sort of, genetic uber and underclass. And likewise, too, we don't want to overgeneralize about particular racial and ethnic categories or particular, people with disabilities, right? Have also, individualized concerns within generalized categories. But I think to some extent, just because we want to make sure that we have diversity, we end up relying on these proxies. And again, what we need to do is we need to be careful to make sure that we're not overgeneralizing and saying that we're speaking in terms of probabilities and things and saying, well, we know that this is not, all individuals in this given population, but, there's an overrepresentation. Or as Meyer was saying, I think that, maybe a more scientific way would be to rely on clustering in terms of ancestry, where that is something that has less social meaning. But I think that to totally remove the social meaning would disserve us, because it does have significance, when you're talking about particular categories that have experienced disparities and discrimination.
So in the interest of inclusion, what I'm going to suggest is that we have people who have comments or questions to make them, and then I'll give Maya and Jessica the last word. How does that sound?
We'll just sit.
So, yeah.
So I have a question for solve the problem. Are you proposing then that each clinical trial has to have different arms based on race, one for whites, one for African Americans, one for Asians? For everybody else, you exclude one group and then they're going to start screening. We know some therapies work in different races differently. Some are more effective than others. And then you have to take into account the return on investment for these pharma companies and the cost of running these trials. And maybe, working in a specific population is not going to be as beneficial to that company as it would be in the larger, broader population. So
which is why I don't think that we should require all the precision medicine projects to have, all of them have to be with the exact formula of representation. But I think, again, for the All of Us program, for sure, I think they have to be more aware of that.
Yes.
And it's the commercialization problem, again, the fact that pharmaceutical companies are concerned about the bottom line opposed to actual helping health.
Yeah.
So.
Well, I have a complicated question, too, so maybe just something to think about. And the question is whether we're overprotecting these groups. I think a significant barrier to participation in research for minority communities and those with disabilities is that IRBs and the research community consider them to be vulnerable populations. And obviously they are in certain circumstances. But this, much of the PMI is minimal risk research, particularly that phase where you're just collecting data and trying to develop associations and biobanking and that sort of thing. So the question is, what's the trade-off between adequate protection for individuals in those communities and adequate recruitment? Is it a real trade-off, and should we be thinking about a lower level of protection, frankly, in order to enhance a competing value of inclusiveness?
So I see George over there waving his hand, and there were a couple of hands back there that I want to make sure to get as well.
Be quiet.
So I just had a quick question for Dr. Sabatella, really, which is, this morning we heard Larry Meyer talk about the Million Veterans Project, and I am just intuiting that they must do something in terms of disabled veterans And I'm wondering whether there is something to be learned from the Million Veterans Project in terms of your, emphasis on greater inclusion of disabled people in these sort of larger general population studies.
Is Larry here? He's left? I would have loved to ask him that as well.
A couple of quick ones, and then we've got about.
I wanted to say that actually I think partly what we're missing is the how absolutely difficult it is to it's one thing to have a commitment to be inclusive. It's another thing to actually make it possible for people to participate, depending on what exactly the nature of the barrier is. And I think there's a real issue of resources. I hear actually a lot of lip service on the part of the All of Us program. I don't think they've solved at all, and I don't think they know any better than a lot of us do how to solve the problem of how to actually be inclusive on a whole variety of levels. And frankly, the resources devoted to this are not huge, actually, for the All of Us research program. And you can think about things, too, like that the resources that go into making it possible to get more diversity in terms of who's included come from other things. And as always happens in these sort of, large efforts, there are real competing influences, and people battle over resources and so forth. So it's, I think there's a strong commitment there to and an awareness of what needs to be done. I don't yet really see that there's an awareness of when you're recruiting 1 million people, how you actually do that. In a way that, for example, we know if trust is an issue for people who are underrepresented historically in research, and particularly for some groups given the prior abuses of research that they've experienced, we know that really authenticity, or we think we know anyway, that authenticity and transparency are really important. Direct actual contact and being able to talk to people. But how do you scale that to hundreds of thousands of people? It's, I think, not a small problem to solve at all.
So I'm beginning to have a rationing problem too, but there was a hand back there. Do you want to just comment, and then we'll wrap it up here?
I want to ask a question from a kind of the opposite perspective. It seems like we have limitations of funding to decide whom to include and whom not to. But what if you look at the other perspective, that there are companies that are offering genome sequencing for like $100 or $200? I don't know if, I think they do they look for specific markers. I don't even think they do whole exome sequencing, much less whole genome. And at the same time, some states in the U.S., I think California and a bunch of other states also do genome sequencing for all the prisoners, I think. So it seems like there are a bunch of people who are trying to do this at scale. And looking at the way that a lot of technology has been scaling up, both in terms of the communication stuff as well as the bio side, how much does whole exome or whole genome sequencing cost? And this question goes for all the speakers today, because some of you are talking about all the different technologies and standards and whatnot. And where would be a point in time where this could be part of the medical record, even we don't have the current applications, that at some point we can query federated databases of genome sequences or gene expression data to recruit patients. And then at that point, the government could incentivize studying whether it's sickle cell or cystic fibrosis. At that point, who's responsible for the population can decide to prioritize different types of research but at that time we would have a large federated warehouse of genome sequences available. So where are we in terms of like financially supporting that?
So I'm going to ask both of you to comment on this richness of comments.
So I'll just take the last question or comment. I think that, one of the models in terms of having access to just a ton of genetic or genomic information, we do, I mentioned the newborn blood spots in Texas. So that is an opportunity. If we're serious, right, about getting access to everybody's genetic and genomic information, we do take blood from newborns, and that is a way to have a pretty comprehensive genetic and genomic data bank that would be available to everyone, and we wouldn't confront these kinds of issues. And so I teach a genetics in the law class, and I say, well, one possibility is just, we're all in it, right? We're all in the database, and it's just sort of a given and a part of citizenship. In terms of the cost of sequencing, I can't speak to the cost of analysis, but that one company that I mentioned, I think it was something for $400, and you would get your genome, but you get it on a flash drive, and you have access to the data, but the most useful part is the analysis, right? And so I think it depends on the service that you're being offered. So that's kind of like a very, very low cost, but then, you would have to go out to someone and either do the analysis yourself or find someone to do it for you. And so I think that there's price points. And then of course, for an individual test, for genetic tests, then for a particular condition, that's something, could be $100. You can do that recreational genetic testing for $99. So there's a huge range, I think, of access.
And I'll just respond quickly to Jeff's comment or question about the IRBs. I think there's room for protection, for sure. I don't want to sort of waive the protections altogether, but I do think from studies that I've read that IRB members tend to be on the more conservative side. They have a tendency of underestimating capacity to understand. There is an assumption in both clinical and research settings that when someone is declared legally with legal incapacity, that there is an assumption that that person also cannot really participate in any medical and research decisions, where in fact it's not true. You can have different types of participation. You can have participation in smaller decision making rather than sort of taking care of your whole property, for example. So I think there are ways of doing it, and I think there's been also studies suggesting, for example, that advocates of people with intellectual disabilities can come on board, just as we have a community member joining IRBs. We can also have a person with psychiatric disorders or an advocate for people with intellectual disabilities, or actually advocates themselves participating in those discussions and actually able to mitigate more of those concerns or questions that can come up. So it's just kind of to rethink maybe a little bit some of those decisions. Yeah.
I can make a comment on the cost if you like.
Yeah.
So what it costs us up at the Huntsman Cancer Institute when we send these tests out, it varies between $6,000 to $9,000 a test.
So the comment was on the cost for tests. When the Huntsman sends it out, it's between $6,000 and $9,000 per test.
Yeah, you're fine. We're sort of at the end.
Yeah.
Why don't you come here?
But before.
Anyway, it's about $6,000 to $9,000, but that includes all the interpretation and information that's associated with that test and the cost of sequencing. But I do know of this new collaboration now between Intel and the Broad Institute, and they're working on making basically programmatic analysis available, basically making these machines do the algorithms for coming out with the analysis more programmatically through Intel, and Intel is funding this project with the Broad Institute. So eventually, I think these costs for sequencing is one thing, the analysis is a whole other thing. And when those costs for analysis start to come down because machines are doing it, then it will become more ubiquitous and more people will have access to it. But as far as where we're at now, I think it's a very, very big step to take to get to $9,000. Yeah, you can sequence your genome for $100, but you also have to be careful about who's doing the interpretation and the analysis on it, as well as who's doing the sequencing, because the DNA and those samples can be polluted, and then if there's any sample degradation, then you're not getting the results that you'd like to have based on your genome.
So I want to thank both Jessica and Maya for raising some of the most pressing questions about access.
Thank you, Leslie, and thank you everyone again.
And yes, thank you all.
Yes. Thanks. So that concludes the portion of the proceedings for today. We'll be back tomorrow at 9 o'clock here in the same room. If you are a speaker and if you are going to take the shuttle bus back to the hotel, we'll gather everyone together and walk down. So why don't we just meet here? And we'll walk you down to where the shuttle bus will be. So don't go far. I see people wandering off, but come back if you want to be on the shuttle bus. Okay, anyway, we'll see you all in the morning. Thank you.
Shuttle bus is here? Yes, shuttle bus is here. We'll take you to the hotel.